Chapter 1: The Immune System Architecture. The immune system represents one of biology's most sophisticated networks, a complex array of cells, tissues, and molecular mechanisms that work together to protect the body from an endless variety of threats, while maintaining tolerance to beneficial microorganisms and self-tissues. This remarkable system must distinguish between self and non-self with extraordinary precision, mounting vigorous responses against dangerous pathogens, while avoiding harmful attacks on the body's own tissues and beneficial microbes that comprise the normal microbiota. The immune system's organization reflects millions of years of evolutionary refinement in response to pathogenic challenges, creating a multi-layered defense network that operates at multiple scales, from individual cells to coordinated systemic responses. This system exhibits remarkable properties including specificity, memory, diversity, and self-tolerance that enable it to recognize and respond to a virtually unlimited array of potential threats, while learning from each encounter to mount more effective responses upon re-exposure. Physical and chemical barriers form the immune system's first line of defense, preventing most potential pathogens from establishing infections before they can activate cellular immune responses. The skin provides a formidable physical barrier, reinforced by antimicrobial secretions, and a resident microbiota that competes with potential pathogens for resources and attachment sites. Mucous membranes lining the respiratory, digestive, and urogenital tracts secrete antimicrobial substances including lactoferrin, lysozyme, and secretory antibodies, while employing physical mechanisms such as ciliary action and mucus production to remove potential threats. The complement system exemplifies the sophisticated molecular machinery underlying innate immunity, comprising over 30 proteins that can recognize pathogens directly or enhance the activities of antibodies and other immune components. This cascade system can be activated through multiple pathways including direct pathogen recognition, antibody binding, or tissue damage signals, ultimately leading to pathogen destruction through membrane perforation, enhanced phagocytosis, and inflammatory responses that recruit additional immune effectors to sites of infection. Cellular innate immunity involves multiple specialized cell types that can recognize and respond to pathogens within hours of initial exposure, providing immediate protection, while activating adaptive immune responses that develop over days to weeks. Neutrophils represent the most abundant circulating immune cells, rapidly migrating to infection sites where they engulf pathogens, release antimicrobial substances, and form neutrophil extracellular traps that can immobilize and kill large numbers of microorganisms. Macrophages serve as versatile immune sentinels that combine potent antimicrobial activities with crucial roles in immune regulation and tissue repair. These long-lived cells can adopt different functional states ranging from classically activated antimicrobial phenotypes to alternatively activated repair and regulatory phenotypes, demonstrating the immune system's ability to adjust responses based on local conditions and the nature of encountered threats. Dendritic cells function as professional antigen-presenting cells that bridge innate and adaptive immunity by capturing antigens at sites of infection and presenting them to naive T cells in secondary lymphoid organs. This process of antigen presentation is crucial for initiating adaptive immune responses and determining whether those responses will be tolerogenic or immunogenic, making dendritic cells key regulators of immune system activation and tolerance. Natural killer cells provide innate immunity against virus-infected cells and tumor cells through sophisticated recognition systems that detect the loss of normal self-markers or the presence of stress-induced ligands. These cells can rapidly eliminate abnormal cells without prior sensitization, providing crucial protection against intracellular pathogens and malignant transformation while avoiding damage to healthy tissues. Pattern recognition receptors enable innate immune cells to distinguish between self and microbial molecules through evolutionary conserved recognition systems that detect pathogen-associated molecular patterns and damage-associated molecular patterns. Toll-like receptors, the most extensively studied pattern recognition receptors, can recognize diverse microbial components including bacterial lipopolysaccharides, viral nucleic acids and fungal cell wall components, triggering signaling cascades that activate antimicrobial responses and inflammatory reactions. The inflammatory response represents a coordinated reaction to tissue damage or infection that involves vascular changes, cellular recruitment, and molecular mediator release designed to eliminate threats and promote healing. While inflammation is generally protective, excessive or persistent inflammatory responses can cause tissue damage and contribute to various diseases, highlighting the importance of inflammatory regulation and resolution. Lymphoid organs provide specialized environments for immune cell development, activation, and interaction, creating the anatomical framework necessary for effective immune responses. Primary lymphoid organs, including the bone marrow and thymus, support immune cell development and education, while secondary lymphoid organs, including lymph nodes, spleen, and mucosa-associated lymphoid tissues, provide sites for antigen presentation and immune response initiation. The bone marrow serves as the primary site of hematopoiesis, generating all blood cell lineages including immune cells throughout life. This process involves complex regulatory networks that balance the production of different cell types, while maintaining stem cell populations capable of responding to increased demands during infection or other stress conditions. The thymus provides the specialized environment necessary for T-cell development and selection, ensuring that mature T-cells can recognize self-MHC molecules while eliminating those that react strongly with self-antigens. This process of positive and negative selection is crucial for establishing self-tolerance, while maintaining a diverse T-cell repertoire capable of recognizing foreign antigens. Secondary lymphoid organs create organized microenvironments that facilitate encounters between antigen-presenting cells and lymphocytes, enabling the initiation of adaptive immune responses. The architecture of these organs, with distinct B-cell and T-cell zones connected by specialized trafficking pathways, optimizes the probability of productive interactions between cells carrying complementary recognition specificities. Immune cell trafficking involves sophisticated navigation systems that guide immune cells to appropriate locations based on chemokine gradients, adhesion molecule interactions, and tissue-specific homing signals. This trafficking is essential for immune surveillance, response initiation, and the establishment of immune memory in tissues throughout the body. Chapter 2: Adaptive Immunity and Lymphocyte Function. Adaptive immunity provides the immune system's most sophisticated and powerful defense mechanisms through lymphocytes that can recognize virtually any molecular structure while maintaining exquisite specificity and developing long-lasting memory of previous encounters. This system's remarkable capabilities emerge from complex processes of gene rearrangement, selection, activation, and differentiation that create diverse populations of B and T lymphocytes capable of mounting highly specific responses against particular antigens. B lymphocytes serve as the primary effectors of humoral immunity, producing antibodies that can neutralize pathogens, facilitate their elimination by other immune mechanisms, and provide long-lasting protection against reinfection. The development of B cells involves extensive gene rearrangement processes that create an enormous diversity of antigen recognition specificities from a limited number of gene segments, enabling the immune system to recognize essentially any molecular structure that might be encountered. The process of VD-J recombination generates antibody diversity through the random recombination of variable diversity and joining gene segments that encode the antigen-binding regions of antibody molecules. This combinatorial process, enhanced by junctional diversity and somatic hypermutation, can theoretically generate over 10 to the power of 11 different antibody specificities from fewer than 1,000 gene segments, far exceeding the number of different antigens likely to be encountered during a lifetime. Antibody structure reflects the dual requirements for antigen recognition and effector function, with variable regions that determine antigen specificity linked to constant regions that mediate interactions with other immune system components. The Y-shaped structure of antibodies enables simultaneous binding to multiple copies of the same antigen, while presenting constant regions in optimal orientations for complement activation or binding to cellular Fc receptors. Class switching allows B cells to produce antibodies with different effector functions while maintaining the same antigen specificity, enabling appropriate responses to different types of threats. IgM antibodies provide early responses with high avidity for repetitive antigens. IgG antibodies offer versatile systemic protection with efficient tissue penetration. IgA antibodies protect mucosal surfaces, and IgE antibodies mediate allergic responses and antiparasitic immunity. Affinity maturation improves antibody binding strength through somatic hypermutation and selection processes that occur in specialized microenvironments within secondary lymphoid organs. B cells undergo rapid proliferation and mutation in germinal centers, with selection favoring variants that bind antigen more strongly, resulting in progressive improvements in antibody affinity over the course of immune responses. Memory B cells provide the cellular basis for long-lasting humoral immunity, maintaining the ability to rapidly produce high-affinity antibodies upon re-encounter with previously recognized antigens. These cells can persist for decades and respond more quickly and effectively than naive B cells, providing the foundation for vaccine-induced protection and natural immunity following infection. T lymphocytes mediate cellular immunity through diverse effector functions including direct cytotoxicity, helper functions that enhance other immune responses, and regulatory activities that maintain immune homeostasis. T-cell development in the thymus involves positive selection for cells that can recognize self-MHC molecules, and negative selection against cells that react too strongly with self-antigens, creating a mature T-cell population that is both self-MHC restricted and self-tolerant. CD8+ T cells, also known as cytotoxic T lymphocytes, specialize in recognizing and eliminating cells that display foreign antigens in association with MHC class I molecules. These cells provide crucial protection against intracellular pathogens, including viruses and some bacteria, by directly killing infected cells before pathogens can complete their replication cycles, while also contributing to tumor immune surveillance. CD4+ T cells serve diverse helper functions that enhance and coordinate immune responses through the production of cytokines and direct cellular interactions. Different subsets of CD4+ T cells specialize in promoting different types of immune responses, with Th1 cells enhancing cellular immunity against intracellular pathogens, Th2 cells promoting humoral immunity and antiparasitic responses, and Th17 cells coordinating inflammatory responses against extracellular bacteria and fungi. Regulatory T cells maintain immune tolerance and prevent excessive immune responses through suppressive mechanisms that can inhibit the activation and effector functions of other immune cells. These cells are essential for preventing autoimmune diseases and limiting immune responses to prevent tissue damage, while also maintaining tolerance to beneficial microorganisms and environmental antigens. Major histocompatibility complex molecules play central roles in T-cell recognition by presenting peptide fragments to T-cell receptors, enabling the immune system to monitor the protein content of cells throughout the body. MHC class I molecules present intracellular peptides to CD8+ T cells, allowing detection of infected or abnormal cells, while MHC class II molecules present extracellular peptides to CD4+ T cells, enabling responses to environmental antigens. Antigen processing and presentation involve complex intracellular pathways that generate peptide fragments appropriate for MHC presentation, while maintaining the information content necessary for immune recognition. The proteasome degrades intracellular proteins for MHC class I presentation, while endocytic pathways process extracellular antigens for MHC class II presentation, creating two parallel systems that survey different cellular compartments. T-cell activation requires multiple signals that ensure responses occur only when appropriate, preventing autoimmune reactions while enabling effective responses to genuine threats. The first signal involves T-cell receptor recognition of peptide MHC complexes, while the second signal requires costimulatory interactions between accessory molecules on T cells and antigen-presenting cells, creating a two-step verification system that prevents inappropriate activation. Immunological memory represents one of adaptive immunity's most important features, enabling faster and more effective responses to previously encountered antigens through populations of memory lymphocytes that persist long after initial immune responses have resolved. Memory cells can respond more rapidly than naive cells, and often require less stringent activation conditions, providing enhanced protection against reinfection. The generation of immunological memory involves complex selection processes that determine which activated lymphocytes will differentiate into memory cells rather than short-lived effector cells. These decisions are influenced by factors including antigen persistence, inflammatory signals, and cellular interactions that collectively determine the magnitude and duration of memory responses. Central and effector memory cells represent functionally distinct memory populations that provide protection through different mechanisms. Central memory cells patrol secondary lymphoid organs and can rapidly proliferate upon antigen re-encounter, while effector memory cells circulate through peripheral tissues and can immediately perform effector functions without extensive proliferation. Tissue-resident memory cells represent a recently discovered memory population that remains permanently stationed in peripheral tissues, providing rapid local protection against reinfection. These cells can respond within hours of pathogen re-encounter, and may be particularly important for protection against pathogens that typically cause localized infections. Chapter 3: Immune Recognition and Response Mechanisms. The immune system's ability to recognize and respond appropriately to an enormous diversity of potential threats, while maintaining tolerance to self and beneficial microorganisms, depends on sophisticated molecular recognition systems that have evolved to distinguish subtle differences between molecular structures. These recognition mechanisms operate at multiple levels, from pattern recognition receptors that detect broad classes of pathogens, to highly specific antigen receptors that can discriminate between closely related molecular variants. Molecular mimicry presents one of the most challenging recognition problems for the immune system, as pathogens often evolve surface structures that resemble host molecules to evade immune detection. This evolutionary arms race has driven the development of recognition systems that can detect pathogen-specific features while avoiding cross-reactivity with self-antigens, though imperfect discrimination can sometimes lead to autoimmune responses when immune recognition of pathogens extends to structurally similar self-molecules. Danger signals provide crucial context for immune recognition by indicating when immune responses are appropriate, helping to distinguish between harmless environmental exposures and genuine threats requiring immune intervention. Damage-associated molecular patterns released from dying or stressed cells can trigger inflammatory responses even in the absence of infectious agents, while pathogen-associated molecular patterns provide direct evidence of microbial presence. The complement system demonstrates how molecular recognition can be amplified through cascade reactions that convert initial recognition events into powerful effector responses. Classical pathway activation involves antibody binding to antigens, while alternative pathway activation can occur through direct recognition of pathogen surfaces, and lectin pathway activation involves recognition of carbohydrate patterns typical of microbial surfaces. Antibody-antigen interactions exemplify the specificity achievable through molecular recognition, with binding affinities that can exceed those of most enzyme-substrate interactions, while maintaining selectivity that can distinguish between antigens differing by a single amino acid or chemical modification. The three-dimensional structure of antibody binding sites creates complementary surfaces that optimize interactions with specific antigenic determinants while excluding non-specific binding partners. Cross-reactivity in antibody recognition can provide broad protection against related pathogens, but can also lead to undesired reactions with self-antigens or beneficial microorganisms. Understanding cross-reactivity patterns is crucial for vaccine design and for predicting potential autoimmune complications of immune responses, while also explaining how exposure to one pathogen can sometimes provide protection against related organisms. T-cell receptor recognition involves even more stringent specificity requirements than antibody recognition, as T-cells must simultaneously recognize both antigenic peptides and self-MHC molecules while avoiding recognition of self-peptide MHC complexes. This dual recognition requirement creates a system that is both highly specific and inherently self-restricted, ensuring that T-cell responses are directed against foreign antigens presented by self cells. Superantigens represent a special class of antigens that can bypass normal T-cell specificity requirements by binding simultaneously to MHC class II molecules and T-cell receptors outside their normal antigen binding sites. This binding can activate large numbers of T-cells regardless of their antigen specificity, leading to massive cytokine release and potentially fatal toxic shock syndrome, demonstrating how pathogens can exploit immune recognition systems. Immunodominance describes the phenomenon where immune responses focus on particular antigenic determinants within complex antigens, even when many other potential recognition sites are available. This focusing of responses may reflect structural accessibility, processing efficiency, or competition between different antigenic determinants for presentation, and understanding immunodominance is crucial for designing effective vaccines that elicit responses against the most protective antigenic sites. Immune evasion strategies employed by pathogens demonstrate the evolutionary pressure on recognition systems and highlight potential vulnerabilities that can be exploited therapeutically. Antigenic variation allows pathogens to change their surface structures faster than immune memory can adapt, while molecular mimicry can prevent recognition altogether, and some pathogens actively interfere with antigen processing and presentation pathways. Cytokine networks provide communication systems that coordinate immune responses across different cell types and anatomical locations, enabling the integration of local immune events into systemic responses appropriate for the nature and magnitude of encountered threats. Different cytokines can promote different types of immune responses, with type 1 cytokines enhancing cellular immunity, type 2 cytokines promoting humoral immunity, and type 3 cytokines coordinating inflammatory responses. Interferons represent a particularly important class of cytokines that provide early protection against viral infections by inducing antiviral states in target cells and enhancing immune recognition of infected cells. Type 1 interferons are produced rapidly by infected cells and can protect neighboring cells from infection, while type 2 interferon produced by immune cells enhances cellular immunity and macrophage activation. Chemokines serve as navigation signals that guide immune cell movement to appropriate locations during immune responses, creating concentration gradients that attract specific cell types to sites where their functions are needed. Different chemokines can attract different cell types, enabling the sequential recruitment of immune effectors appropriate for different phases of immune responses. Immune checkpoints provide regulatory mechanisms that prevent excessive immune responses, while maintaining the ability to respond effectively to genuine threats. These systems involve inhibitory receptors and ligands that can dampen immune activation when appropriate, preventing tissue damage from overly vigorous immune responses while maintaining responsiveness to new threats. Tolerance mechanisms ensure that immune recognition systems do not attack self-tissues or beneficial microorganisms, involving both central tolerance established during lymphocyte development and peripheral tolerance maintained throughout life. Central tolerance eliminates lymphocytes with strong self-reactivity during their development, while peripheral tolerance involves multiple mechanisms including regulatory T cells, anergy and deletion that prevent autoimmune responses. Immune privilege exists in certain anatomical sites, where immune responses are limited to prevent tissue damage that could compromise essential functions. The eye, brain and reproductive organs maintain immune privilege through various mechanisms including physical barriers, immunosuppressive factors, and limited lymphatic drainage that collectively minimize immune responses while maintaining protection against serious threats. Oral tolerance represents a specialized form of immune tolerance that prevents harmful immune responses to dietary antigens and beneficial gut microorganisms, while maintaining the ability to respond to pathogenic organisms. This tolerance involves specialized dendritic cell populations, regulatory T cells, and unique cytokine environments that promote tolerogenic rather than immunogenic responses to ingested antigens. Immune aging involves changes in immune recognition and response capabilities that occur with advancing age, including reduced responsiveness to new antigens, increased susceptibility to infections, and elevated risks of autoimmune diseases. Understanding immune aging is crucial for developing strategies to maintain immune function throughout life, and for designing vaccines and therapies appropriate for elderly populations. Chapter 4: Immune System Disorders and Dysfunction. Immune system disorders encompass a broad spectrum of conditions that result from either insufficient immune responses that leave individuals vulnerable to infections and malignancies, or excessive immune responses that cause tissue damage through allergic reactions, autoimmune diseases, or chronic inflammation. Understanding these disorders provides crucial insights into normal immune function, while highlighting the delicate balance required for optimal immune system performance. Primary immunodeficiencies represent inherited disorders that affect immune system development or function, creating increased susceptibility to infections that may be life-threatening without appropriate treatment. These conditions can affect different components of the immune system, from severe combined immunodeficiency that eliminates both T and B cell function, to more selective defects that affect specific immune pathways or cell types. Severe combined immunodeficiency exemplifies the devastating consequences of complete immune system failure, with affected infants developing serious infections within the first months of life due to the absence of functional T cells and often B cells as well. Multiple genetic defects can cause SCID, including deficiencies in enzymes required for lymphocyte development, defects in cytokine receptors essential for immune cell survival, and mutations affecting VDJ recombination processes. Antibody deficiencies represent the most common class of primary immunodeficiencies, causing increased susceptibility to bacterial infections due to impaired humoral immunity. X-linked agammaglobulinemia results from defects in B-cell development that prevent antibody production entirely, while common variable immunodeficiency involves more complex defects that reduce antibody production and function without completely eliminating B cells. Complement deficiencies illustrate how defects in individual immune system components can create specific patterns of susceptibility, with different complement deficiencies causing increased risks for different types of infections. Early complement component deficiencies increase susceptibility to encapsulated bacteria and autoimmune diseases, while late complement component deficiencies particularly increase risks for Neisseria infections. Phagocyte disorders, affect the ability of neutrophils and macrophages to eliminate pathogens through defects in cell migration, pathogen recognition, or antimicrobial mechanisms. Chronic granulomatous disease results from defects in NADPH oxidase that prevent the generation of reactive oxygen species essential for bacterial killing, leading to recurrent infections with catalase-positive bacteria and fungi. Secondary immunodeficiencies result from external factors that impair immune function, including malnutrition, aging, stress, medications, and infections that damage immune cells or interfere with immune responses. HIV infection represents the most significant secondary immunodeficiency, progressively destroying CD4+ T cells and creating susceptibility to opportunistic infections and malignancies that define AIDS. Autoimmune diseases occur when immune responses are inappropriately directed against self-tissues, causing chronic inflammation and tissue damage that can affect virtually any organ system. These diseases result from failures in tolerance mechanisms that normally prevent immune recognition of self-antigens, though the specific triggers that initiate autoimmune responses often remain unknown. Systemic lupus erythematosus exemplifies multisystem autoimmune disease, with antibodies against nuclear antigens causing inflammation in joints, skin, kidneys, and other organs. The diversity of autoantibodies in lupus reflects widespread breakdown of tolerance mechanisms, while the chronic nature of the disease demonstrates how autoimmune responses can become self-perpetuating through tissue damage and inflammatory amplification. Rheumatoid arthritis demonstrates how autoimmune responses can focus on particular tissues, with inflammatory responses primarily affecting synovial joints, while causing systemic manifestations that reflect the chronic inflammatory state. The progression from initial joint inflammation to bone and cartilage destruction illustrates how immune responses intended to eliminate threats can cause irreversible tissue damage when inappropriately directed. Type 1 diabetes results from autoimmune destruction of pancreatic beta cells that produce insulin, demonstrating how autoimmune responses can eliminate specific cell types essential for normal physiological function. The long preclinical phase during which autoantibodies are detectable before clinical diabetes develops suggests opportunities for intervention if reliable predictive markers and effective prevention strategies can be developed. Multiple sclerosis involves autoimmune responses against central nervous system components, particularly myelin proteins that insulate nerve fibers and enable rapid electrical conduction. The demyelination and inflammation that characterize multiple sclerosis can cause diverse neurological symptoms depending on the locations of immune attacks, while the relapsing-remitting pattern typical of early disease reflects the episodic nature of autoimmune flares. Allergic diseases result from inappropriate immune responses to harmless environmental antigens, with IgE antibodies and mast cell degranulation, creating symptoms ranging from mild allergic rhinitis to life-threatening anaphylaxis. The increase in allergic diseases in developed countries may reflect reduced exposure to microorganisms during early life that affects immune system development and tolerance. Asthma involves chronic airway inflammation driven by allergic responses to inhaled antigens, with eosinophils, Th2 cells, and inflammatory mediators creating bronchial hyperresponsiveness and airway remodeling that can persist even when allergen exposure is minimized. The complex interactions between genetic susceptibility, environmental exposures, and immune responses make asthma a paradigmatic example of multifactorial disease. Food allergies demonstrate how immune responses to dietary proteins can override normal oral tolerance mechanisms, creating risks for severe reactions upon ingestion of specific foods. The increasing prevalence of food allergies, particularly in children, suggests that environmental or lifestyle factors may be affecting tolerance development, though the specific mechanisms remain unclear. Chronic inflammatory diseases, encompass conditions where dysregulated immune responses create persistent inflammation that damages tissues over time. Inflammatory bowel disease involves inappropriate immune responses to gut microorganisms, while chronic obstructive pulmonary disease involves inflammatory responses to inhaled irritants that progressively destroy lung tissue. Transplant rejection represents a specialized form of immune dysfunction, where normal immune recognition of foreign tissue antigens prevents successful organ transplantation without immunosuppressive therapy. Understanding the mechanisms of rejection has led to improved immunosuppressive protocols, though the balance between preventing rejection and maintaining immunity against infections and malignancies remains challenging. Graft versus host disease can occur following bone marrow transplantation when donor immune cells recognize recipient tissues as foreign and mount immune responses that can damage multiple organ systems. This complication illustrates how immune responses that would normally be protective can become pathological when immune cells are placed in foreign environments. Tumor immunology reveals how immune surveillance mechanisms can both prevent and promote cancer development, with immune responses capable of eliminating transformed cells, while chronic inflammation can promote tumor growth and progression. Understanding these dual roles of immunity in cancer has led to new therapeutic approaches that enhance anti-tumor immune responses while limiting pro-tumor inflammatory processes. Immunosenescence describes the changes in immune function that occur with aging, including reduced responses to new antigens, increased susceptibility to infections, elevated rates of autoimmune diseases, and decreased vaccine efficacy. These changes may result from thymic involution, chronic inflammation, and cellular senescence that collectively impair immune system function in elderly individuals. Chapter 5: Vaccines, Therapeutics and Future Directions. The application of immunological principles to prevent and treat disease has produced some of medicine's greatest successes, while continuing to drive innovative approaches to challenging health problems. Vaccination remains one of the most cost-effective public health interventions ever developed, preventing millions of deaths annually, while demonstrating the power of harnessing natural immune mechanisms for disease prevention. Vaccine development involves complex considerations of antigen selection, formulation, delivery, and safety that must balance effectiveness with acceptability across diverse populations. Traditional vaccine approaches, including live attenuated vaccines, inactivated vaccines, and subunit vaccines, each offer different advantages and limitations, while newer approaches, including DNA vaccines, viral vector vaccines, and mRNA vaccines, are expanding the range of diseases that can be prevented through vaccination. Live attenuated vaccines provide robust and durable immunity by using weakened versions of pathogens that can replicate sufficiently to stimulate strong immune responses without causing disease in healthy individuals. These vaccines often provide lifelong immunity after a single dose and stimulate both cellular and humoral immune responses, though they may pose risks for immunocompromised individuals and require careful maintenance of the cold chain. Inactivated vaccines use killed pathogens or pathogen components that cannot replicate, but retain sufficient antigenic structure to stimulate protective immune responses. While generally safer than live vaccines, these preparations often require multiple doses and adjuvants to achieve adequate immunity, and they may not stimulate cellular immunity as effectively as live vaccines. Subunit vaccines focus immune responses on specific pathogen components known to be protective antigens, potentially reducing unwanted reactions while concentrating responses on the most important targets. Protein subunit vaccines, polysaccharide vaccines, and conjugate vaccines represent different approaches to presenting protective antigens in forms that stimulate appropriate immune responses. Adjuvants enhance vaccine effectiveness by stimulating innate immune responses that promote stronger and more durable adaptive immunity. These substances can include aluminum salts, oil emulsions, or molecular adjuvants that activate specific pattern recognition receptors, enabling reduced antigen doses while improving immune responses, particularly in populations with diminished immune responsiveness. Herd immunity describes how vaccination of a sufficient proportion of a population can protect unvaccinated individuals by reducing pathogen transmission, providing indirect protection for those who cannot be vaccinated due to medical contraindications or immune deficiencies. Achieving herd immunity requires vaccination coverage levels that vary depending on pathogen transmissibility and vaccine effectiveness. Immunotherapy represents a rapidly expanding field that harnesses or modifies immune responses to treat diseases including cancer, autoimmune disorders, and allergies. These approaches can involve enhancing beneficial immune responses, suppressing harmful ones, or redirecting immune recognition toward desired targets. Cancer immunotherapy has emerged as one of the most promising areas of immunological application, with approaches ranging from checkpoint inhibitors that unleash existing anti-tumor immunity to adoptive cell therapies that genetically modify patient immune cells to recognize tumor antigens. These treatments have produced remarkable responses in some patients with previously untreatable cancers, though responses are often limited to subsets of patients. Checkpoint inhibitors work by blocking inhibitory signals that limit T-cell responses, enabling more vigorous anti-tumor immunity in patients whose immune systems have recognized tumor antigens but are being held in check by regulatory mechanisms. Antibodies against PD-1, PD-L1 and CTLA-4 have shown efficacy against multiple cancer types, though they can also cause autoimmune side effects by reducing normal tolerance mechanisms. CAR T-cell therapy involves genetically modifying patient T-cells to express chimeric antigen receptors that can recognize tumor antigens directly without requiring MHC presentation. These engineered cells can produce dramatic responses against hematologic malignancies, though extending this approach to solid tumors and managing associated toxicities remains significant challenges. Monoclonal antibodies represent versatile therapeutic tools that can be designed to target virtually any cell surface molecule, enabling precise interference with specific immune pathways or cellular functions. These agents have revolutionized treatment for autoimmune diseases, cancers, and other conditions, while providing models for developing new targeted therapies. Immunosuppressive therapy aims to reduce harmful immune responses in autoimmune diseases, transplant recipients, and other conditions where excessive immunity causes tissue damage. These approaches must balance suppression of pathological responses with maintenance of protective immunity against infections and malignancies. Biologics represent a growing class of therapeutics that include antibodies, cytokines, and other immune system components designed to modulate specific immune pathways. These agents often provide more targeted effects than traditional small molecule drugs, but may require specialized manufacturing, storage, and administration procedures. Precision medicine approaches in immunology involve tailoring treatments based on individual immune system characteristics, genetic factors, and biomarkers that predict treatment responses. These approaches may enable more effective therapies with fewer side effects by matching treatments to patients most likely to benefit. Microbiome research has revealed complex interactions between immune system development and function, and the trillions of microorganisms that colonize human bodies. Understanding these interactions may lead to new therapeutic approaches that modify microbial communities to promote immune health and treat immune-related diseases. Regenerative immunology explores how immune responses affect tissue repair and regeneration, with potential applications for enhancing healing, promoting organ regeneration, and treating degenerative diseases. These approaches may involve modulating inflammatory responses, enhancing tissue-specific stem cell function, or directing immune responses toward repair rather than destruction. Systems immunology applies computational approaches to understanding immune system complexity by integrating data from multiple levels of analysis including genomics, transcriptomics, proteomics, and metabolomics. These approaches may reveal new therapeutic targets and enable better prediction of treatment responses. Global health immunology addresses the unique challenges of preventing and treating immune-related diseases in resource-limited settings, including the development of heat-stable vaccines, point-of-care diagnostic tests, and cost-effective treatment strategies appropriate for diverse populations and environments. Aging and immunity research seeks to understand how immune function changes with age, and to develop interventions that can maintain immune competence throughout life. These efforts may lead to improved vaccines for elderly populations, treatments for immunosenescence, and strategies for healthy aging. Artificial intelligence applications in immunology include drug discovery, biomarker identification, and treatment optimization, using machine learning approaches that can identify patterns in complex immunological data that may not be apparent through traditional analysis methods. Ethical considerations in immunology include ensuring equitable access to vaccines and immunotherapies, managing the risks and benefits of immune interventions, and addressing public concerns about vaccine safety while maintaining scientific integrity in the face of misinformation. The future of immunology promises continued advances in understanding immune system complexity, while developing increasingly sophisticated approaches to preventing and treating diseases through immune system modulation. These advances will likely require interdisciplinary collaboration among immunologists, engineers, computational scientists, and other specialists to address complex challenges in immune system biology and therapeutic application. Chapter 1: The Nature of Cancer and Cellular Transformation. Cancer represents one of the most complex and formidable challenges in modern biology and medicine, characterized by the fundamental breakdown of normal cellular control mechanisms that maintain tissue homeostasis and organismal integrity. At its essence, cancer is a disease of cellular behavior where individual cells escape the regulatory constraints that normally govern their growth, division, and death, leading to uncontrolled proliferation and the potential to invade distant tissues. This transformation from normal to malignant cell behavior involves the accumulation of genetic and epigenetic alterations that progressively disrupt the molecular machinery responsible for maintaining cellular order and cooperation within multicellular organisms. The hallmarks of cancer, as originally articulated by Douglas Hanahan and Robert Weinberg, provide a conceptual framework for understanding how normal cells become transformed into cancer cells through the acquisition of specific capabilities that enable malignant behavior. These hallmarks include sustaining proliferative signaling, evading growth suppressors, resisting cell death, enabling replicative immortality, inducing angiogenesis, and activating invasion and metastasis. More recent updates to this framework have included reprogramming energy metabolism, evading immune destruction, genome instability and mutation, and tumor-promoting inflammation as additional enabling characteristics that support cancer development and progression. Cellular transformation represents a multi-step process where normal cells progressively acquire the molecular alterations necessary for malignant behavior. This process, often termed the adenoma-carcinoma sequence in epithelial cancers, involves the sequential accumulation of mutations in genes that control critical cellular functions including cell cycle regulation, DNA repair, apoptosis, and cell adhesion. The transformation process typically requires multiple genetic hits, occurring over extended periods, explaining why cancer incidence increases dramatically with age as more time becomes available for the accumulation of oncogenic mutations. The genetic basis of cancer involves alterations in three major classes of genes that normally function to maintain cellular homeostasis and prevent malignant transformation. Oncogenes represent genes that, when activated or overexpressed, promote cellular proliferation and survival in ways that can contribute to cancer development. These genes often encode proteins involved in growth factor signaling, cell cycle progression or apoptosis inhibition, and their normal cellular functions become corrupted in cancer cells to drive inappropriate cellular behaviors. Tumor suppressor genes represent the second major class of cancer-related genes, functioning normally to prevent uncontrolled cellular proliferation and maintain genomic stability. The loss of tumor suppressor function through mutation, deletion, or epigenetic silencing removes critical barriers to cellular transformation and allows cells to bypass normal growth control mechanisms. The p53 gene, often called the guardian of the genome, exemplifies the crucial role of tumor suppressors in preventing cancer by detecting DNA damage and either promoting repair or triggering cell death when damage is irreparable. DNA repair genes constitute the third major category of cancer-related genes, functioning to maintain genomic stability by detecting and correcting various types of DNA damage that occur spontaneously or result from environmental exposures. Defects in DNA repair mechanisms lead to increased mutation rates and genomic instability that accelerate the accumulation of oncogenic alterations. Hereditary cancer syndromes often result from inherited mutations in DNA repair genes, demonstrating the critical importance of these pathways in preventing cancer development. The concept of clonal evolution provides a fundamental framework for understanding how cancers develop and progress over time through the gradual accumulation of genetic alterations that confer selective advantages to individual cells within growing tumors. Cancer development begins with a single cell that acquires an initial oncogenic mutation, leading to clonal expansion of that altered cell population. Subsequent mutations in individual cells within this clone can provide additional growth advantages, leading to subclonal expansion and the gradual development of increasingly malignant cellular populations. This evolutionary process continues throughout cancer development and progression, with individual tumor cells competing for resources and space, while accumulating additional mutations that may enhance their survival and proliferative capabilities. The heterogeneity that results from this ongoing evolutionary process creates significant challenges for cancer treatment, as different cells within the same tumor may respond differently to therapeutic interventions, and resistant subclones may emerge to repopulate tumors following treatment. Epigenetic alterations represent an increasingly recognized component of cancer development that involves changes in gene expression patterns without corresponding changes in DNA sequence. These alterations include modifications to DNA methylation patterns, histone modifications, and chromatin structure that can silence tumor suppressor genes or activate oncogenes in ways that contribute to malignant transformation. Epigenetic changes can be inherited through cell divisions and may be influenced by environmental factors, providing additional mechanisms for cancer development beyond simple genetic mutations. The tumor microenvironment plays crucial roles in cancer development and progression by providing the cellular and molecular context within which cancer cells exist and interact with surrounding normal tissues. The microenvironment includes various cell types such as fibroblasts, immune cells, endothelial cells, and others that can either support or suppress cancer growth depending on the specific circumstances and signals they receive. Understanding these complex interactions has revealed that cancer is not simply a disease of individual rogue cells, but rather involves complex ecosystems of interacting cellular populations. Inflammation represents a critical component of the tumor microenvironment that can both promote and suppress cancer development through complex mechanisms that depend on the type, duration, and context of inflammatory responses. Chronic inflammation can promote cancer development by creating tissue environments that favor cellular proliferation, angiogenesis, and genetic instability, while also suppressing immune surveillance that normally eliminates potentially malignant cells. Conversely, acute inflammatory responses can enhance immune recognition and elimination of cancer cells, highlighting the complex dual roles that inflammation plays in cancer biology. Cancer stem cells represent a subpopulation of cancer cells that possess enhanced self-renewal capability and the ability to generate the heterogeneous lineages of cancer cells that comprise tumors. These cells may be responsible for tumor initiation, maintenance and recurrence following treatment, as they can remain dormant during therapy and subsequently repopulate tumors when treatment is discontinued. The cancer stem cell hypothesis has important implications for understanding cancer biology and developing therapeutic strategies that target these critical cell populations. Metabolic reprogramming emerges as a fundamental characteristic of cancer cells that enables them to meet the increased biosynthetic and energetic demands associated with rapid proliferation. Cancer cells often exhibit altered glucose metabolism, characterized by increased glycolysis even in the presence of oxygen, a phenomenon known as the Warburg effect. This metabolic shift provides cancer cells with the building blocks necessary for rapid proliferation, while also creating acidic tissue environments that may facilitate invasion and suppress immune function. The relationship between aging and cancer reflects the shared molecular mechanisms that underlie both processes, including DNA damage accumulation, cellular senescence, and immune dysfunction. While aging is the single greatest risk factor for cancer development, the relationship between these processes is complex, with some aspects of aging potentially promoting cancer development while others may provide protection. Understanding these relationships has important implications for cancer prevention and treatment, particularly as populations age and cancer incidence continues to increase. Hereditary cancer syndromes provide valuable insights into the fundamental mechanisms of cancer development by revealing how inherited mutations in specific genes dramatically increase cancer risk. These syndromes demonstrate the importance of particular genes and pathways in preventing cancer, while also illustrating how genetic counseling and testing can enable risk assessment and prevention strategies for individuals with inherited cancer predisposition. The study of hereditary cancer syndromes has led to important discoveries about cancer biology that have informed understanding of sporadic cancers as well. Chapter 2: Molecular Mechanisms of Cancer Development. The molecular mechanisms underlying cancer development involve complex networks of cellular pathways that normally function to maintain tissue homeostasis but become corrupted in cancer cells to drive malignant behavior. Understanding these mechanisms requires examining how specific molecular alterations affect cellular functions, including proliferation, survival, DNA repair, and interaction with surrounding tissues. The progressive accumulation of these molecular defects creates increasingly abnormal cellular behavior that ultimately manifests as the clinical and pathological features that define cancer. Modern molecular biology techniques have revealed the extraordinary complexity of these mechanisms, while also identifying common themes and pathways that are disrupted across different cancer types. Cell cycle regulation represents one of the most fundamental cellular processes that becomes dysregulated in cancer, involving the complex molecular machinery that controls when and how often cells divide. Normal cells tightly regulate their progression through the cell cycle phases, including the G1 phase where cells grow and prepare for DNA synthesis, the S phase where DNA replication occurs, the G2 phase where cells prepare for division, and the M phase where actual cell division takes place. Multiple checkpoints throughout the cell cycle ensure that cells only proceed to the next phase when they are properly prepared and when external conditions are appropriate for division. The molecular machinery controlling cell cycle progression includes cyclins and cyclin-dependent kinases that form complexes responsible for driving cells through different phases of the cycle. Cyclins are regulatory proteins whose levels fluctuate throughout the cell cycle, while cyclin-dependent kinases are enzymes that phosphorylate target proteins to promote cell cycle progression. The activity of these complexes is controlled by multiple regulatory mechanisms, including inhibitory proteins, phosphatases, and proteolytic degradation that ensure cell cycle progression occurs only under appropriate conditions. Cancer cells frequently exhibit dysregulation of cell cycle control mechanisms that allows them to bypass normal growth constraints and proliferate inappropriately. Oncogenes such as MYC, cyclin D1 and cyclin E can drive excessive cell cycle progression by promoting S-phase entry even when external growth signals are absent or when DNA damage is present. Conversely, tumor suppressor genes such as RB and P53 normally function to prevent inappropriate cell cycle progression but are frequently inactivated in cancer cells, removing critical barriers to uncontrolled proliferation. The P53 pathway represents one of the most important tumor suppressor mechanisms in human cells, functioning as a cellular stress sensor that can detect various forms of cellular damage including DNA breaks, hypoxia, oncogene activation, and ribosomal stress. When activated, P53 can trigger multiple cellular responses including cell cycle arrest to allow time for repair, apoptosis to eliminate severely damaged cells, or senescence to permanently remove cells from the proliferative pool. The P53 protein is often called the guardian of the genome because of its central role in maintaining genomic stability and preventing the propagation of potentially oncogenic mutations. DNA damage response pathways represent crucial cellular mechanisms that detect and repair various types of DNA lesions that occur spontaneously or result from environmental exposures. These pathways include base excision repair for small base modifications, nucleotide excision repair for bulky DNA adducts, mismatch repair for replication errors, and homologous recombination and non-homologous end joining for double-strand breaks. Defects in DNA repair mechanisms are found in many cancers and contribute to the genomic instability that characterizes malignant cells. The concept of synthetic lethality has emerged as an important principle for understanding cancer vulnerabilities and developing targeted therapies. Synthetic lethality occurs when the combination of defects in two genes is lethal, while defects in either gene alone are tolerable. Cancer cells that have lost one DNA repair pathway may become dependent on alternative repair mechanisms, creating therapeutic opportunities to selectively kill cancer cells by targeting the remaining repair pathway while sparing normal cells that retain both pathways. Apoptosis, or programmed cell death, represents a fundamental cellular process that normally eliminates damaged or unnecessary cells to maintain tissue homeostasis. This process involves a complex cascade of molecular events that culminate in cellular self-destruction through the activation of caspases enzymes that degrade cellular components in an orderly fashion. Normal cells can undergo apoptosis in response to various stimuli including DNA damage, growth factor withdrawal, or developmental signals that indicate the cell is no longer needed. Cancer cells frequently acquire resistance to apoptosis through various mechanisms that allow them to survive under conditions that would normally trigger cell death. These mechanisms include overexpression of anti-apoptotic proteins such as BCL2 family members, loss of pro-apoptotic proteins such as BAX and BAC, and mutations in P53 that prevent DNA damage-induced apoptosis. The ability to evade apoptosis is considered one of the enabling characteristics of cancer that allows malignant cells to survive and proliferate despite accumulating significant cellular damage. Telomere biology plays crucial roles in cancer development through its effects on cellular aging and genomic stability. Telomeres are protective DNA protein structures at chromosome ends that shorten with each cell division, eventually triggering senescence or cell death when they become critically short. Normal somatic cells have limited replicative potential due to telomere shortening, but cancer cells frequently reactivate telomerase enzyme activity that extends telomeres and enables unlimited proliferative potential. The reactivation of telomerase in cancer cells represents a crucial step in malignant transformation that enables the unlimited proliferative potential necessary for tumor growth and progression. However, this process must be carefully balanced, as excessive telomerase activity can promote cancer development, while insufficient activity may limit tumor growth. Understanding telomere biology has led to therapeutic strategies aimed at targeting telomerase activity in cancer cells while preserving normal stem cell function. Growth factor signaling pathways represent another major category of molecular mechanisms that become dysregulated in cancer cells. Normal cells depend on external growth signals to proliferate, but cancer cells often acquire the ability to generate their own growth signals or become hypersensitive to normal growth factor levels. Oncogenes such as EGFR, HER2, and RAS are frequently overexpressed or mutated in cancers, leading to excessive growth factor signaling that drives inappropriate cellular proliferation. The PI3K-ACT-mTOR pathway represents one of the most commonly altered signaling networks in human cancers, regulating multiple cellular processes including metabolism, growth, survival, and proliferation. Activation of this pathway promotes cellular survival and growth while suppressing apoptosis, making it an attractive target for cancer cells. Mutations or amplifications affecting components of this pathway are found in a large proportion of human cancers and have led to the development of targeted therapies aimed at inhibiting pathway activity. RAS proteins represent a family of small GTPases that play central roles in growth factor signaling and are among the most frequently mutated genes in human cancers. RAS proteins normally cycle between active GTP-bound and inactive GDP-bound states, but cancer-associated mutations often lock RAS in the active state, leading to continuous growth and survival signals. Despite decades of research, directly targeting mutant RAS proteins has proven challenging, though recent advances have led to the development of specific inhibitors for certain RAS mutations. Angiogenesis, the formation of new blood vessels, represents a crucial process that enables tumor growth beyond the size limitations imposed by diffusion of nutrients and oxygen. Normal tissues tightly regulate angiogenesis to ensure adequate blood supply, while preventing excessive vessel formation, but cancer cells often acquire the ability to stimulate angiogenesis through the production of pro-angiogenic factors such as VEGF. This acquired capability enables tumors to grow to large sizes and provides pathways for metastatic spread to distant sites. The tumor microenvironment plays increasingly recognized roles in cancer development and progression through its effects on cellular behavior and therapeutic responses. The microenvironment includes various cell types such as cancer-associated fibroblasts, immune cells, endothelial cells, and others that interact with cancer cells through direct contact and secreted factors. These interactions can either promote or suppress cancer progression, depending on the specific context and signals involved. Cancer-associated fibroblasts represent activated mesenchymal cells that can promote cancer progression through the secretion of growth factors, cytokines, and extracellular matrix components that support tumor growth, angiogenesis, and invasion. These cells can be recruited and activated by cancer cells through various signaling mechanisms, and may represent important therapeutic targets for disrupting tumor-supporting microenvironments. Immune evasion represents a crucial capability that cancer cells must acquire to survive and proliferate in the face of immune surveillance mechanisms that normally eliminate abnormal cells. Cancer cells employ various strategies to evade immune recognition and destruction, including downregulation of antigen presentation machinery, secretion of immunosuppressive factors, and recruitment of regulatory immune cell populations that suppress anti-tumor immune responses. Chapter 3: Cancer Progression and Metastasis. Cancer progression represents the process by which localized tumors evolve to become increasingly aggressive and ultimately acquire the ability to invade surrounding tissues, and establish secondary tumors at distant sites through metastasis. This progression involves fundamental changes in cellular behavior that enable cancer cells to overcome the physical and biological barriers that normally confine cells to their tissues of origin. Understanding the mechanisms underlying cancer progression and metastasis is crucial for developing effective therapeutic strategies, as metastatic disease represents the primary cause of cancer-related mortality and presents significant challenges for treatment due to the disseminated nature of advanced cancer. The transition from benign to malignant tumor behavior involves the acquisition of invasive capabilities that enable cancer cells to breach tissue boundaries and enter surrounding normal tissues. This process requires significant changes in cellular adhesion, motility, and interaction with extracellular matrix components that normally maintain tissue architecture and prevent inappropriate cellular movement. Cancer cells must downregulate adhesion molecules that normally keep cells anchored within tissues, while upregulating proteins that facilitate cellular movement and matrix degradation. Cell adhesion molecules, particularly E-cadherin, play crucial roles in maintaining epithelial tissue integrity by forming adherens junctions that link adjacent cells together and anchor them to the underlying basement membrane. The loss of E-cadherin expression or function is a hallmark of epithelial mesenchymal transition, a process where epithelial cells acquire mesenchymal characteristics including increased motility and invasive capacity. This transition is regulated by various transcription factors including snail, slug, twist and zeb that suppress epithelial gene expression while promoting mesenchymal gene expression. The extracellular matrix represents a complex network of proteins and glycosaminoglycans that provides structural support for tissues while also regulating cellular behavior through biochemical and physical signals. Normal cells are exquisitely sensitive to matrix composition and organization, with inappropriate matrix contacts often triggering apoptosis through a process called anoikos. Cancer cells must overcome this matrix dependence to survive outside their normal tissue environment, and must also acquire the ability to degrade matrix barriers that impede cellular movement. Matrix metalloproteinases represent a family of enzymes that degrade various extracellular matrix components and play crucial roles in enabling cancer cell invasion. These enzymes are normally tightly regulated to prevent inappropriate matrix degradation, but cancer cells often overexpress metalloproteinases or dysregulate their inhibitors to create permissive environments for invasion. The balance between metalloproteinases and their tissue inhibitors determines the extent of matrix remodeling and influences cancer cell invasive capacity. The basement membrane represents a specialized extracellular matrix structure that separates epithelial tissues from underlying stroma and normally serves as a barrier to cancer cell invasion. Breach of the basement membrane is considered a defining feature of invasive carcinoma and requires coordinated degradation of basement membrane components including collagen IV, laminin and proteoglycans. Cancer cells accomplish basement membrane invasion through the secretion of specific enzymes and the recruitment of stromal cells that assist in matrix degradation. Angiogenesis becomes increasingly important during cancer progression, as growing tumors require adequate blood supply to sustain continued growth and provide routes for metastatic dissemination. The angiogenic switch represents a crucial transition where tumors acquire the ability to stimulate new blood vessel formation through the production of pro-angiogenic factors such as VEGF, FGF, and PDGF. This switch is regulated by the balance between pro-angiogenic and anti-angiogenic signals and can be influenced by hypoxia, metabolic stress and genetic alterations that affect angiogenic signaling pathways. Tumor angiogenesis differs significantly from normal physiological angiogenesis in terms of vessel structure, function and regulation. Tumor blood vessels are often abnormal in structure, with irregular branching patterns, variable diameters, and increased permeability that reflects the dysregulated nature of tumor angiogenesis. These structural abnormalities can affect drug delivery to tumors and may contribute to the hypoxic and acidic microenvironments that characterize many solid tumors. The metastatic cascade represents a complex multi-step process that begins with local invasion and culminates in the establishment of secondary tumors at distant sites. This process requires cancer cells to successfully complete multiple sequential steps, including invasion of surrounding tissues, entry into blood or lymphatic vessels, intravasation, survival in the circulation, arrest at distant sites, exit from vessels into target tissues, extravasation, and establishment of secondary tumors, colonization. The low efficiency of this process means that only a small fraction of cancer cells that enter the circulation successfully establish metastatic tumors. Circulating tumor cells represent cancer cells that have entered the bloodstream and can potentially establish metastatic tumors at distant sites. The detection and characterization of circulating tumor cells has become an active area of research, with potential applications for monitoring cancer progression, assessing treatment responses, and understanding metastatic mechanisms. However, the rarity of these cells and their heterogeneity presents significant technical challenges for their isolation and analysis. The concept of metastatic organotropism describes the observation that different cancer types tend to metastasize to specific organ sites in predictable patterns. Breast cancer commonly metastasizes to bone, liver, lung, and brain, while prostate cancer shows a strong preference for bone metastasis, and colorectal cancer frequently metastasizes to the liver. This organ-specific pattern of metastasis reflects complex interactions between cancer cell properties and the microenvironments of different target organs. The seed and soil hypothesis, originally proposed by Stephen Paget in 1889, suggests that successful metastasis requires compatible interactions between cancer cells, the seed, and the microenvironment of target organs, the soil. This concept has been refined by modern research, showing that cancer cells must be able to survive in the target organ microenvironment, adapt to local growth conditions, and establish supportive interactions with resident cell populations to successfully establish metastatic tumors. Pre-metastatic niche formation represents a recently discovered mechanism where primary tumors can condition distant sites to become more receptive to metastatic colonization before cancer cells actually arrive. This process involves the secretion of factors by primary tumors that recruit bone marrow-derived cells and modify the microenvironment of distant organs to create conditions favorable for subsequent metastatic establishment. Understanding pre-metastatic niche formation may provide opportunities for preventing metastasis by targeting these preparatory mechanisms. Dormancy represents a poorly understood aspect of cancer progression, where individual cancer cells or small clusters of cells can remain in a quiescent state for extended periods before subsequently reactivating to form clinically detectable metastases. Cancer cell dormancy may occur at primary tumor sites, in circulation, or at distant tissue sites, and appears to involve cell cycle arrest mechanisms that maintain cell viability while preventing proliferation. The signals that control entry into and exit from dormancy are not well understood, but may involve interactions with the microenvironment, immune surveillance, and intrinsic cellular programs. Epithelial mesenchymal transition plays crucial roles in cancer progression by endowing cancer cells with enhanced motility, invasive capacity, and resistance to apoptosis that facilitate metastatic spread. This transition involves the coordinated downregulation of epithelial characteristics, such as cell-cell adhesion and apical basal polarity, along with the upregulation of mesenchymal features, including increased motility and matrix-degrading enzyme production. The reverse process, mesenchymal epithelial transition, may be important for metastatic colonization, by enabling circulating cancer cells to re-establish epithelial characteristics necessary for proliferation and tumor formation. Cancer stem cells may play particularly important roles in metastasis due to their enhanced self-renewal capacity, resistance to therapy, and ability to regenerate heterogeneous tumor populations. These cells may be preferentially capable of surviving the stresses of the metastatic process and establishing secondary tumors at distant sites. The cancer stem cell hypothesis has important implications for understanding why metastatic disease is often resistant to conventional therapies that may eliminate the bulk of cancer cells while sparing the stem cell populations capable of regenerating tumors. Immune evasion becomes increasingly important during cancer progression, as cancer cells must avoid immune surveillance while spreading to new tissue sites where they may encounter different immune microenvironments. Cancer cells employ various strategies to evade immune recognition, including downregulation of major histocompatibility complex molecules, secretion of immunosuppressive factors, and induction of immune tolerance through regulatory T-cell recruitment. The ability to evade immune surveillance may be particularly important for successful metastatic colonization in immunologically active organs. Metabolic reprogramming continues to evolve during cancer progression, as cancer cells adapt to the changing metabolic demands and microenvironmental conditions encountered during metastatic spread. Cancer cells must adapt their metabolism to survive in circulation, where nutrients and oxygen may be limited, and must subsequently adapt to the metabolic conditions present in target organs that may differ significantly from the primary tumor site. This metabolic flexibility may represent both a vulnerability that can be targeted therapeutically and a mechanism that enables successful metastatic colonization. Chapter 4: Cancer Heterogeneity and Evolution. Cancer heterogeneity represents one of the most challenging aspects of cancer biology, encompassing the remarkable diversity observed within individual tumors, among tumors of the same type, and across different patients. This heterogeneity manifests at multiple levels, including genetic, epigenetic, transcriptomic, proteomic, and phenotypic differences that create complex mosaics of cellular populations within tumors. Understanding cancer heterogeneity is crucial for developing effective therapeutic strategies, because this diversity underlies many of the challenges associated with cancer treatment, including therapeutic resistance, disease recurrence, and the difficulty of predicting treatment responses. The evolutionary nature of cancer means that heterogeneity is not static, but rather continues to evolve throughout tumor development and treatment, creating a moving target for therapeutic intervention. Intratumor heterogeneity describes the remarkable diversity that exists among cancer cells within individual tumors, where different regions of the same tumor can contain genetically and phenotypically distinct cancer cell populations. This heterogeneity arises through the ongoing process of clonal evolution, where individual cancer cells acquire new mutations over time, leading to the development of genetically distinct subclones within the growing tumor mass. Single-cell sequencing technologies have revealed the extraordinary extent of this intratumor heterogeneity, showing that individual tumors can contain hundreds or thousands of genetically distinct cancer cells. The spatial organization of intratumor heterogeneity reflects the clonal evolution process, where related cancer cell populations tend to be located in proximity to each other based on their shared evolutionary history. However, cancer cells can also migrate within tumors, leading to the intermixing of different clonal populations and the creation of complex spatial patterns of genetic diversity. This spatial heterogeneity has important implications for tumor sampling and diagnosis, as single biopsies may not accurately represent the full genetic diversity present within tumors. Temporal heterogeneity describes how tumor composition changes over time as cancer cells continue to evolve and as selective pressures from therapy, immune responses, and microenvironmental factors shape clonal populations. This evolutionary process means that tumor characteristics at diagnosis may differ significantly from those present at recurrence or progression, creating challenges for treatment strategies based on initial tumor characteristics. Longitudinal studies using serial biopsies or circulating tumor DNA analysis have revealed the dynamic nature of cancer evolution and the emergence of new clonal populations during treatment. The cancer evolutionary process is driven by several key factors including mutation rate, selection pressure, population size, and genetic drift that together determine the rate and direction of evolutionary change. Cancer cells typically exhibit increased mutation rates due to defective DNA repair mechanisms, creating abundant genetic variation upon which natural selection can act. The large population sizes of cancer cell populations mean that even mutations with small fitness effects can be efficiently selected, while genetic drift effects are minimized except in very small populations such as those present during metastatic colonization. Clonal evolution models provide frameworks for understanding how cancer develops and progresses through the accumulation of genetic alterations over time. Linear evolution models suggest that cancers progress through ordered sequences of genetic changes, similar to the adenoma-carcinoma sequence described for colorectal cancer. However, branching evolution models propose that cancer development involves the divergent evolution of multiple subclones that may coexist within tumors and follow different evolutionary trajectories. Neutral evolution represents an alternative model where much of the genetic diversity observed in cancers results from random genetic drift rather than natural selection. Under this model, many mutations that accumulate in cancer cells are neither beneficial nor harmful, but rather neutral with respect to fitness, leading to the random accumulation of genetic diversity over time. Distinguishing between neutral and selected mutations has important implications for understanding cancer biology and identifying therapeutic targets. Driver and passenger mutations represent a crucial distinction in cancer genetics, where driver mutations confer selective advantages that contribute to cancer development and progression, while passenger mutations are present due to chance and do not contribute to malignant behavior. Identifying driver mutations among the thousands of mutations present in individual cancers is a major challenge that requires large-scale genomic studies and sophisticated computational approaches to distinguish functional alterations from random background mutations. Tumor suppressor gene inactivation often requires multiple hits according to the Knudsen two-hit hypothesis, where both copies of a tumor suppressor gene must be inactivated to eliminate its protective function. This model helps explain why cancer development typically requires multiple genetic alterations, and why individuals with inherited mutations in tumor suppressor genes have dramatically increased cancer risk. However, recent research has revealed that haploinsufficiency, where partial loss of tumor suppressor function contributes to cancer risk, may be more common than originally thought. Oncogene addiction describes the phenomenon where cancer cells become dependent on particular oncogenes for survival and proliferation, even though normal cells do not require these genes. This dependence creates therapeutic opportunities for selectively targeting cancer cells by inhibiting specific oncogenes while sparing normal cells. However, cancer cells can often overcome oncogene addiction by activating alternative pathways or acquiring resistance mutations, limiting the durability of targeted therapies. Therapeutic resistance represents one of the most significant consequences of cancer heterogeneity and evolution, where cancer cells develop mechanisms to survive and proliferate despite the presence of anti-cancer therapies. Resistance can arise through various mechanisms including target mutation, pathway activation, drug efflux, metabolic reprogramming, and phenotypic switching that enable cancer cells to evade therapeutic effects. The presence of resistant subclones within tumors before treatment begins means that some cancers may be intrinsically resistant to therapy from the outset. Acquired resistance develops during treatment, as cancer cells evolve mechanisms to overcome therapeutic pressure through the selection of pre-existing resistant cells or the development of new resistance mechanisms. This process can occur rapidly, particularly with targeted therapies that create strong selective pressure for resistance development. Understanding the mechanisms of acquired resistance has led to strategies for combination therapy and sequential treatment regimens designed to prevent or delay resistance development. Minimal residual disease represents the small populations of cancer cells that may persist after treatment and serve as sources of disease recurrence. These residual cancer cell populations may be present at the primary tumor site, in regional lymph nodes, or at distant metastatic sites, and may exhibit different characteristics from the bulk tumor population. Detection and characterization of minimal residual disease has become an important area of research, with potential applications for monitoring treatment responses and predicting disease recurrence. Cancer stem cells may play particularly important roles in therapeutic resistance due to their enhanced DNA repair capacity, increased drug efflux capability, and altered metabolic characteristics that make them less susceptible to conventional therapies. These cells may survive treatment that eliminates the bulk of cancer cells, and subsequently repopulate tumors with resistant cancer cell populations. Targeting cancer stem cells requires different therapeutic approaches than those effective against rapidly proliferating cancer cells. Liquid biopsies represent emerging technologies for monitoring cancer evolution and heterogeneity through analysis of circulating tumor DNA, circulating tumor cells, and other blood-based biomarkers that reflect the molecular characteristics of cancer cells throughout the body. These approaches offer the potential for real-time monitoring of cancer evolution and therapeutic responses without the need for invasive tissue biopsies. However, technical challenges remain in detecting and analyzing the small amounts of tumor-derived material present in blood samples. Personalized medicine approaches seek to tailor cancer treatment strategies to the specific molecular characteristics of individual tumors, taking into account the genetic alterations present in each patient's cancer. This approach requires comprehensive tumor profiling to identify actionable mutations that can be targeted with specific therapies. However, intratumor heterogeneity means that single biopsies may not capture the full spectrum of genetic alterations present within tumors, potentially leading to incomplete or inaccurate treatment selection. Precision oncology represents the clinical application of cancer genomics to guide treatment decisions based on the molecular characteristics of individual tumors. This approach has led to remarkable successes in treating cancers with specific genetic alterations, such as HER2-positive breast cancers, EGFR mutant lung cancers, and tumors with microsatellite instability. However, most cancer patients do not have tumors with clear actionable mutations, and those who do often develop resistance to targeted therapies over time. Evolutionary therapy represents an emerging treatment paradigm that applies evolutionary principles to cancer treatment, recognizing that conventional maximum tolerated dose approaches may actually accelerate resistance development by creating strong selective pressure for resistant clones. Adaptive therapy strategies involve modulating treatment intensity based on tumor response to maintain sensitive cancer cell populations that can compete with resistant cells and slow resistance development. Combination therapy approaches seek to overcome cancer heterogeneity and prevent resistance development by simultaneously targeting multiple pathways or mechanisms that cancer cells require for survival and proliferation. These approaches can involve combinations of targeted therapies, conventional chemotherapy and targeted agents, or immunotherapy and other treatment modalities. However, determining optimal combination strategies requires understanding the complex interactions among different therapeutic mechanisms and their effects on cancer cell populations. Chapter 5: Cancer Prevention, Detection and Treatment. Cancer prevention represents the most effective approach for reducing cancer burden, encompassing strategies that reduce cancer incidence through the elimination or modification of risk factors, early detection of precancerous conditions, and intervention before malignant transformation occurs. Prevention strategies operate at multiple levels, including primary prevention that removes carcinogenic exposures, secondary prevention that detects and treats precancerous lesions, and tertiary prevention that prevents recurrence in cancer survivors. Understanding the multifactorial nature of cancer causation is crucial for developing effective prevention strategies, as most cancers result from complex interactions among genetic susceptibility, environmental exposures, and lifestyle factors that accumulate over extended periods. Primary prevention focuses on reducing exposure to known carcinogens and modifying lifestyle factors that influence cancer risk. Tobacco use represents the single most important preventable cause of cancer, contributing to approximately 1/3 of cancer deaths and increasing risk for multiple cancer types including lung, bladder, cervix, kidney, and numerous others. Tobacco cessation programs and policies that reduce tobacco use have demonstrated remarkable success in reducing cancer incidence and mortality, with lung cancer rates declining significantly in populations where smoking rates have decreased. Infectious agents contribute to approximately 15 to 20% of cancers worldwide, with viral, bacterial, and parasitic infections serving as important modifiable risk factors for cancer development. Human papillomavirus infections cause virtually all cervical cancers and many other anogenital and head and neck cancers, while hepatitis B and C viruses are major causes of liver cancer. Helicobacter pylori bacteria increase stomach cancer risk, and various other infectious agents contribute to cancer development through chronic inflammation, immune suppression, or direct oncogenic effects. Vaccination represents one of the most effective cancer prevention strategies available, with vaccines against hepatitis B virus and human papillomavirus demonstrating remarkable success in reducing cancer incidence in vaccinated populations. The hepatitis B vaccine has dramatically reduced liver cancer rates in countries with widespread vaccination programs, while HPV vaccines are expected to substantially reduce cervical cancer incidence as vaccinated cohorts reach ages of peak cancer risk. These successes have stimulated research into vaccines against other cancer-causing infectious agents. Diet and nutrition play complex roles in cancer development, with various dietary components potentially increasing or decreasing cancer risk through effects on inflammation, oxidative stress, hormone levels, and other biological mechanisms. High consumption of processed meats and red meat has been associated with increased colorectal cancer risk, while diets rich in fruits and vegetables may provide protection against various cancer types. However, the relationship between specific dietary components and cancer risk remains an active area of research, with ongoing controversies about optimal dietary recommendations. Physical activity emerges as one of the most consistent protective factors against cancer development, with regular exercise reducing risk for multiple cancer types including breast, colon, endometrial, and others. The mechanisms underlying exercise's protective effects may include improved immune function, reduced inflammation, better hormone regulation, and maintenance of healthy body weight. Physical activity recommendations for cancer prevention generally align with those for overall health promotion, emphasizing regular moderate to vigorous exercise as part of healthy lifestyle patterns. Environmental and occupational carcinogen exposure represents important targets for cancer prevention through regulatory policies and workplace safety measures. Asbestos exposure has been dramatically reduced in many countries through regulations that have led to corresponding decreases in mesothelioma and lung cancer rates. Air pollution, particularly fine particulate matter, represents an increasingly recognized cancer risk factor that requires policy interventions at local, national, and international levels to reduce population exposures. Cancer screening programs aim to detect cancer or precancerous conditions before symptoms develop, when treatment is most likely to be effective and cure rates are highest. Effective screening requires tests that can accurately distinguish between individuals with and without cancer or precancerous conditions, while being acceptable to the target population and cost-effective for healthcare systems. Successful screening programs have been established for cervical, breast, and colorectal cancers, leading to significant reductions in mortality from these diseases. Cervical cancer screening represents one of the most successful cancer prevention programs, with Pap smear testing enabling detection and treatment of precancerous cervical lesions before they progress to invasive cancer. The introduction of HPV testing has further improved screening effectiveness by identifying women at highest risk for developing cervical cancer. Organized screening programs with systematic invitation and follow-up procedures have achieved greater population impact than opportunistic screening approaches. Mammography screening for breast cancer has demonstrated effectiveness in reducing breast cancer mortality in multiple randomized controlled trials, though debates continue about optimal screening ages, intervals, and target populations. Digital mammography and tomosynthesis have improved screening performance by reducing false positive rates and increasing cancer detection sensitivity. However, concerns about overdiagnosis and the psychological and economic costs of false positive results continue to influence screening recommendations. Colorectal cancer screening offers multiple effective approaches including fecal occult blood testing, sigmoidoscopy, and colonoscopy that can detect both early-stage cancers and precancerous polyps. Colonoscopy screening with polypectomy has been particularly effective because it enables simultaneous detection and treatment of precancerous lesions, preventing progression to invasive cancer. Newer screening approaches, including stool DNA testing and CT colonography, provide alternative options for individuals who cannot or will not undergo traditional screening methods. Early detection strategies beyond population screening include symptom awareness programs that educate individuals about cancer warning signs and promote prompt medical evaluation of suspicious symptoms. These approaches are particularly important for cancers that lack effective screening tests or occur in populations not covered by screening programs. Symptom awareness campaigns have shown modest success in promoting earlier diagnosis of various cancer types, though their impact on mortality outcomes requires further evaluation. Diagnostic approaches for suspected cancer cases involve coordinated evaluation using imaging studies, laboratory tests, and tissue sampling to establish accurate diagnoses and staging information that guide treatment decisions. Modern diagnostic imaging, including CT, MRI, and PET scanning, provides detailed anatomical and functional information about tumor extent and spread that is crucial for treatment planning. Molecular diagnostics increasingly supplement traditional pathological evaluation to provide information about tumor biology that influences treatment selection. Liquid biopsies represent emerging diagnostic approaches that analyze circulating tumor DNA, circulating tumor cells, or other blood-based biomarkers to detect cancer, monitor treatment responses, and identify disease recurrence. These approaches offer the potential for less invasive cancer detection and monitoring, though technical challenges remain in achieving sufficient sensitivity and specificity for clinical application. Research continues into applications of liquid biopsies for early cancer detection, treatment selection, and surveillance of cancer survivors. Cancer treatment has evolved dramatically with the development of targeted therapies, immunotherapies, and precision medicine approaches that complement traditional surgery, chemotherapy, and radiation therapy. Treatment strategies increasingly involve multidisciplinary teams that coordinate different treatment modalities to optimize outcomes while minimizing treatment-related toxicity. Personalized treatment approaches based on tumor molecular characteristics and patient factors are becoming standard practice for many cancer types. Surgical treatment remains a cornerstone of cancer therapy, particularly for localized tumors where complete surgical removal can achieve cure. Advances in surgical techniques including minimally invasive approaches, robotic surgery, and improved reconstruction methods have reduced surgical morbidity while maintaining or improving oncological outcomes. Surgical treatment planning increasingly incorporates detailed imaging and molecular information to optimize surgical approaches and determine appropriate adjuvant therapy. Radiation therapy has been revolutionized by technological advances that enable precise targeting of tumors while sparing surrounding normal tissues. Intensity-modulated radiation therapy, stereotactic radiosurgery, and proton beam therapy represent sophisticated approaches that can deliver high doses to tumors while minimizing radiation exposure to critical normal structures. Integration of imaging guidance and adaptive planning enables real-time adjustment of radiation delivery based on tumor and normal tissue changes during treatment. Systemic therapy encompasses chemotherapy, targeted therapy, immunotherapy, and hormonal therapy approaches that can reach cancer cells throughout the body and are particularly important for treating metastatic disease. Traditional chemotherapy agents target rapidly dividing cells but lack specificity for cancer cells, leading to significant toxicity. Targeted therapies designed to inhibit specific molecular pathways required by cancer cells offer improved selectivity and reduced toxicity for patients whose tumors harbor appropriate molecular targets. Immunotherapy has emerged as a revolutionary cancer treatment approach that harnesses the immune system's natural ability to recognize and eliminate cancer cells. Checkpoint inhibitor antibodies that block inhibitory signals on immune cells have demonstrated remarkable efficacy in treating various cancer types, leading to durable responses and improved survival in some patients. CAR T-cell therapy represents another immunotherapy approach where patient immune cells are genetically modified to enhance their anti-cancer activity before being reinfused into the patient. Treatment resistance remains a major challenge in cancer therapy, with most advanced cancers eventually developing resistance to initial treatments through the evolution of resistant cancer cell populations. Understanding resistance mechanisms has led to strategies for combination therapy, sequential treatment regimens, and monitoring approaches that can detect resistance development before clinical progression becomes apparent. Overcoming resistance often requires switching to alternative treatment approaches or combining multiple therapies that target different resistance mechanisms. Survivorship care has become increasingly important, as cancer survival rates have improved and more individuals are living with and beyond cancer. Cancer survivors face ongoing risks of disease recurrence, treatment-related late effects, and secondary cancers that require long-term monitoring and intervention. Comprehensive survivorship care plans address medical surveillance, management of treatment-related complications, health promotion, and psychosocial support to optimize long-term outcomes and quality of life for cancer survivors. Chapter 1: The Cell as the Basic Unit of Life. The cell stands as the fundamental unit of life, representing the smallest organized structure that exhibits all the characteristics that define living systems. This remarkable biological entity serves as both the building block, from which all organisms are constructed, and the basic functional unit that carries out the essential processes of life including metabolism, growth, reproduction, and response to environmental stimuli. The discovery and study of cells has revolutionized our understanding of biology, revealing that despite the incredible diversity of life forms on Earth, from microscopic bacteria to towering redwood trees and complex multicellular organisms like humans, all share this common cellular foundation that unites all living things under a single theoretical framework. The cell theory, formulated in the 19th century through the pioneering work of Matthias Schleiden, Theodor Schwann, and Rudolf Virchow, established three fundamental principles that remain central to modern biology. First, all living organisms are composed of one or more cells, establishing the cell as the basic structural unit of life. Second, the cell is the basic functional unit of life, meaning that all vital processes occur within cellular boundaries and depend on cellular mechanisms. Third, all cells arise from pre-existing cells through cell division, rejecting the notion of spontaneous generation and establishing the continuity of cellular life across generations. The remarkable unity underlying cellular organization becomes apparent when examining the fundamental features shared by all cells, regardless of their origin or complexity. Every cell is bounded by a plasma membrane that separates the internal cellular environment from the external world, while controlling the exchange of materials and information between these compartments. This selective permeability enables cells to maintain distinct internal conditions that are essential for proper cellular function, while remaining responsive to environmental changes and signals. All cells contain genetic material in the form of DNA that stores the information necessary for cellular structure and function, though the organization of this genetic material varies significantly between different cell types. Prokaryotic cells found in bacteria and archaea maintain their DNA in a nucleoid region that lacks a surrounding membrane, while eukaryotic cells enclose their DNA within a membrane-bound nucleus that provides additional levels of organization and regulation. Despite these organizational differences, the fundamental role of DNA as the repository of hereditary information remains constant across all cellular life. The cytoplasm represents another universal cellular feature, consisting of the gel-like matrix that fills the space between the plasma membrane and, in eukaryotic cells, the nuclear envelope. This aqueous medium contains dissolved nutrients, ions and enzymes that facilitate the chemical reactions necessary for cellular metabolism, while providing a supporting framework for cellular structures. The cytoplasm serves as the stage upon which most cellular processes unfold, from protein synthesis and energy production to waste removal and signal transduction. Ribosomes constitute essential cellular machinery found in all cells, responsible for translating genetic information encoded in messenger RNA into the proteins that carry out most cellular functions. These complex ribonucleoprotein particles demonstrate remarkable conservation across all domains of life, though they exhibit structural differences between prokaryotic and eukaryotic cells that reflect their distinct evolutionary histories. The universal presence of ribosomes underscores the central importance of protein synthesis in cellular function and highlights the shared biochemical heritage of all living organisms. The diversity of cellular organization reflects the remarkable evolutionary success of the cellular form of life, which has given rise to an extraordinary array of cell types adapted to virtually every conceivable environmental niche on Earth. Prokaryotic cells represent the earliest and most abundant form of cellular life, exhibiting remarkable metabolic diversity that enables them to thrive in environments ranging from boiling hot springs to frozen Antarctic soils. These cells typically measure between 1 and 5 micrometers in diameter and maintain relatively simple internal organization, yet they carry out all the essential functions of life with remarkable efficiency. Eukaryotic cells emerged later in evolutionary history and exhibit much greater internal complexity through the development of membrane-bound organelles that compartmentalize different cellular functions. This compartmentalization enables eukaryotic cells to achieve much larger sizes than prokaryotic cells while maintaining efficient cellular function through specialized organelles that optimize particular aspects of cellular metabolism. The evolution of eukaryotic cells represents one of the most significant transitions in the history of life, enabling the development of complex multicellular organisms through enhanced cellular cooperation and specialization. The size constraints that govern cellular architecture reflect fundamental physical and chemical principles that influence cellular function and evolution. The surface area to volume ratio represents a critical constraint that affects the ability of cells to exchange materials with their environment, with smaller cells generally exhibiting more efficient exchange rates due to their relatively larger surface areas. This constraint explains why most cells maintain microscopic dimensions, and why larger organisms typically achieve their size through multicellularity rather than through the enlargement of individual cells. Diffusion limitations also impose important constraints on cellular organization and function, as the random movement of molecules through the cytoplasm becomes increasingly inefficient over larger distances. This limitation has driven the evolution of specialized transport mechanisms in larger eukaryotic cells, including active transport systems and intracellular trafficking pathways that ensure efficient distribution of materials throughout the cellular volume. Understanding these physical constraints provides crucial insights into why cells are organized as they are, and how they have evolved to overcome fundamental challenges associated with their microscopic scale. The metabolic requirements of cellular life necessitate sophisticated mechanisms for energy capture, conversion, and utilization that enable cells to maintain their highly organized state, despite the universal tendency toward disorder described by the second law of thermodynamics. Cells accomplish this remarkable feat by coupling energy-releasing reactions with energy-requiring processes, creating metabolic networks that can extract useful energy from environmental sources, while using this energy to drive the synthesis of complex biological molecules and maintain cellular organization. The maintenance of cellular homeostasis represents another fundamental challenge that all cells must address through sophisticated regulatory mechanisms that monitor internal conditions and adjust cellular processes accordingly. These homeostatic mechanisms operate at multiple levels, from individual enzyme regulation to complex signaling networks that coordinate cellular responses to environmental changes. The ability to maintain stable internal conditions while remaining responsive to external signals represents one of the defining characteristics of living systems and distinguishes cells from non-living chemical systems. Cellular reproduction through cell division ensures the continuity of cellular life, while enabling the growth and development of multicellular organisms. The mechanisms of cell division must accurately duplicate all essential cellular components, including the genetic material, while distributing these components equally between daughter cells. This process requires remarkable precision and coordination to ensure that each daughter cell receives the complete set of information and machinery necessary for independent survival and function. The evolutionary perspective on cellular biology reveals how the basic cellular form has been modified and elaborated over billions of years to produce the remarkable diversity of cell types and functions observed in modern organisms. The fossil record provides evidence for the antiquity of cellular life, with microscopic cellular structures preserved in rocks dating back over 3 billion years. This long evolutionary history has provided ample time for the refinement and optimization of cellular processes, resulting in the sophisticated cellular machinery observed in contemporary organisms. Chapter 2: Cellular Architecture and Organization. The internal architecture of cells represents a masterpiece of biological engineering, where complex three-dimensional organization enables the coordination of thousands of simultaneous biochemical reactions within volumes measured in cubic micrometers. This remarkable structural organization varies dramatically between prokaryotic and eukaryotic cells, reflecting different evolutionary solutions to the fundamental challenges of cellular life, including energy production, information storage and processing, waste management, and coordination of cellular activities. Understanding cellular architecture requires examining both the individual components that make up cells and the ways these components are organized and integrated to create functional cellular systems. Prokaryotic cellular architecture emphasizes simplicity and efficiency, with most cellular functions occurring within a single membrane-bounded compartment that lacks the complex internal organization characteristic of eukaryotic cells. The bacterial cytoplasm appears relatively homogeneous under conventional microscopy, but modern research techniques have revealed sophisticated spatial organization that enables efficient cellular function despite the apparent simplicity. The bacterial nucleoid region contains the chromosomal DNA organized by specialized proteins that compact and organize the genetic material without the need for a surrounding nuclear membrane. The plasma membrane of prokaryotic cells serves multiple crucial functions beyond simple compartmentalization, including energy production through respiratory chains embedded in the membrane, nutrient transport through specialized transport proteins, and signal detection through receptor proteins that monitor environmental conditions. The bacterial cell wall provides structural support and protection, while maintaining cell shape and preventing osmotic lysis in hypertonic environments. The composition and structure of bacterial cell walls vary among different bacterial groups, with gram-positive bacteria possessing thick peptidoglycan layers, while gram-negative bacteria have thinner peptidoglycan layers surrounded by additional outer membranes. Eukaryotic cellular architecture achieves remarkable complexity through the evolution of membrane-bound organelles that compartmentalize different cellular functions, while enabling sophisticated coordination among these specialized compartments. The nucleus represents the most prominent organelle in most eukaryotic cells, housing the vast majority of cellular DNA within a double membrane system that controls access to genetic information while enabling complex regulation of gene expression. Nuclear pores regulate the exchange of materials between the nucleus and cytoplasm, ensuring that appropriate molecules can cross the nuclear boundary while maintaining the compartmentalization necessary for proper cellular function. The endoplasmic reticulum extends throughout much of the eukaryotic cytoplasm as an interconnected network of membrane-bound compartments that serve diverse functions in protein synthesis, lipid metabolism, and calcium storage. Rough endoplasmic reticulum, studded with ribosomes on its cytoplasmic surface, specializes in the synthesis and modification of proteins destined for secretion or incorporation into cellular membranes. Smooth endoplasmic reticulum lacks attached ribosomes and focuses on lipid synthesis, drug detoxification, and calcium regulation, demonstrating how membrane specialization enables functional diversity within connected organelle systems. The Golgi apparatus functions as the cellular equivalent of a post office, receiving proteins and other materials from the endoplasmic reticulum, modifying them through various chemical processes, and packaging them for transport to their final destinations within or outside the cell. This organelle exhibits characteristic stacked membrane organization that creates distinct processing compartments, each specialized for particular modification reactions that occur as materials progress through the Golgi stack. The dynamic nature of Golgi organization, with vesicles constantly budding and fusing with different Golgi compartments, enables efficient processing and sorting of cellular products. Mitochondria represent cellular powerhouses that generate most of the ATP required for cellular energy needs through the process of oxidative phosphorylation. These remarkable organelles possess their own DNA and ribosomes, reflecting their evolutionary origin as endosymbiotic bacteria that established permanent residence within eukaryotic cells. The complex internal structure of mitochondria, with extensively folded inner membranes called cristae, maximizes the surface area available for respiratory enzymes while creating distinct compartments that enable efficient energy production. Plant cells exhibit additional architectural complexity through the presence of chloroplasts and other plastids that carry out photosynthesis and other specialized metabolic functions. Chloroplasts, like mitochondria, possess their own genetic systems and exhibit complex internal organization, with elaborate membrane systems, called thylakoids, that house the photosynthetic machinery. The integration of chloroplasts with other cellular systems enables plants to convert light energy into chemical energy, while coordinating photosynthetic activity with other cellular processes. The cytoskeleton provides structural framework and organizational support for eukaryotic cells through networks of protein filaments that extend throughout the cytoplasm. Microfilaments composed of actin provide mechanical support and enable cellular movement through dynamic assembly and disassembly processes. Microtubules formed from tubulin create highways for intracellular transport while also providing structural support and organizing cellular division processes. Intermediate filaments contribute mechanical strength and help maintain cellular shape, particularly in cells subjected to mechanical stress. Membrane systems throughout eukaryotic cells exhibit remarkable diversity and specialization while maintaining fundamental similarities in structure and basic function. All biological membranes share the basic phospholipid bilayer architecture, yet different membranes exhibit distinct lipid compositions, protein contents, and functional capabilities that reflect their specialized roles within cellular organization. The dynamic nature of cellular membranes, with constant fusion and fission events that enable material transport and organelle maintenance, demonstrates the fluid and adaptable nature of cellular architecture. Protein targeting and trafficking systems ensure that newly synthesized proteins reach their appropriate cellular destinations through sophisticated address and delivery mechanisms. Signal sequences embedded within protein structures direct proteins to specific organelles or cellular locations, while specialized transport machinery ensures accurate delivery. This targeting system enables cells to coordinate protein synthesis with functional requirements while maintaining appropriate protein concentrations in different cellular compartments. Vesicular transport systems connect different organelles through the formation, transport, and fusion of membrane-bound vesicles that carry cargo between cellular compartments. These transport systems require precise molecular machinery that can recognize appropriate target membranes, dock vesicles at correct locations, and fuse membranes to deliver cargo while maintaining organelle identity. The specificity and efficiency of vesicular transport enable complex cellular organization while ensuring that materials reach their intended destinations. The endolysosomal system represents a specialized set of organelles responsible for degrading and recycling cellular components through sophisticated digestive processes. Lysosomes contain powerful digestive enzymes that can break down virtually any biological material, while maintaining these enzymes in specialized compartments that protect the rest of the cell from their destructive activity. The coordination between endocytosis, lysosome function, and autophagy demonstrates how cells manage the complex challenge of selective degradation while maintaining essential cellular components. Peroxisomes represent specialized organelles that carry out important metabolic functions, including fatty acid oxidation and hydrogen peroxide detoxification. These organelles demonstrate how cells can create specialized compartments for particular metabolic pathways while maintaining integration with broader cellular metabolism. The ability of peroxisomes to proliferate in response to cellular demands illustrates the dynamic nature of organelle populations and their responsiveness to changing cellular requirements. The spatial organization of cellular components reflects both functional requirements and physical constraints that influence cellular architecture. The positioning of organelles within cells is not random but rather reflects complex interactions among organelles, cytoskeletal elements, and molecular motors that create organized cellular landscapes. This spatial organization enables efficient cellular function while providing flexibility for reorganization in response to changing cellular needs or environmental conditions. Cellular polarity represents another crucial aspect of cellular architecture, where different regions of cells exhibit distinct characteristics and functions. Many cell types establish and maintain asymmetric organization that enables specialized functions such as directed secretion, cellular movement, or tissue formation. The molecular mechanisms that establish and maintain cellular polarity involve complex interactions among membrane components, cytoskeletal elements, and signaling molecules that create stable, yet adaptable asymmetric organization. Chapter 3: Cellular Metabolism and Energy. Cellular metabolism encompasses the vast network of chemical reactions that sustain life by extracting energy from environmental sources, synthesizing essential biological molecules, and maintaining the complex organization that characterizes living systems. This intricate web of biochemical pathways represents one of the most remarkable achievements of biological evolution, enabling cells to capture and transform energy while building and maintaining the sophisticated molecular machinery necessary for cellular function. Understanding cellular metabolism requires examining both the individual chemical reactions that comprise metabolic pathways and the regulatory mechanisms that coordinate these pathways to meet changing cellular demands while maintaining overall metabolic balance. Energy metabolism represents the central challenge that all cells must solve to maintain their highly organized state, in defiance of the universal tendency toward disorder described by thermodynamics. Cells accomplish this remarkable feat by coupling energy-releasing catabolic reactions with energy requiring anabolic processes, creating metabolic networks that can extract useful energy from nutrients, while using this energy to drive biosynthesis and maintain cellular organization. The fundamental currency of cellular energy is adenosine triphosphate, a high-energy molecule that serves as the universal energy carrier in all forms of life. Glycolysis represents one of the most ancient and universal metabolic pathways, breaking down glucose molecules to generate ATP, while producing pyruvate that can be further metabolized through various pathways depending on cellular conditions and oxygen availability. This pathway occurs in the cytoplasm of all cells and involves a series of 10 enzymatic reactions that gradually extract energy from glucose while preserving much of this energy in chemical bonds that can be used for cellular work. The regulation of glycolysis involves multiple control points that enable cells to adjust glucose metabolism in response to energy demands and nutrient availability. The citric acid cycle, also known as the Krebs cycle, represents the central hub of cellular metabolism where the products of carbohydrate, fat, and protein breakdown converge for complete oxidation to carbon dioxide and water. This cyclical pathway occurs within mitochondria in eukaryotic cells and in the cytoplasm of prokaryotic cells, generating reducing equivalents in the form of NADH and FADH2 that carry high-energy electrons to the respiratory chain for ATP production. The citric acid cycle also provides important precursor molecules for biosynthetic pathways, demonstrating the integration between catabolic and anabolic metabolism. Oxidative phosphorylation represents the most efficient mechanism for ATP production, using the energy released during electron transport to pump protons across membranes and create electrochemical gradients that drive ATP synthesis. This process occurs in mitochondria in eukaryotic cells and in the plasma membrane of prokaryotic cells, involving complex molecular machinery that couples electron transport with proton pumping and ATP synthesis. The efficiency of oxidative phosphorylation enables cells to extract maximum energy from nutrients while generating the large amounts of ATP required for cellular processes. Photosynthesis represents the ultimate source of energy for most life on Earth, converting light energy into chemical energy while producing the oxygen that enables aerobic respiration. This complex process occurs in chloroplasts of plants and cyanobacteria, involving light-capturing reactions that generate ATP and reducing power, followed by carbon fixation reactions that incorporate atmospheric carbon dioxide into organic molecules. The integration of photosynthesis with other cellular processes demonstrates how cells can harness environmental energy sources while coordinating energy production with metabolic demands. Lipid metabolism provides both energy storage and structural components essential for cellular function, with fatty acids serving as concentrated energy reserves, while phospholipids and other complex lipids form the structural foundation of cellular membranes. Fatty acid synthesis and degradation involve distinct pathways that are carefully regulated to maintain appropriate cellular lipid levels while responding to changing energy states and membrane requirements. The versatility of lipid metabolism enables cells to adapt to varying nutritional conditions while maintaining essential membrane functions. Amino acid metabolism connects protein synthesis and degradation with central metabolic pathways, providing both building blocks for protein synthesis and alternative energy sources during periods of glucose limitation. The synthesis of amino acids requires complex pathways that often involve multiple regulatory mechanisms to ensure appropriate levels of each amino acid while avoiding wasteful overproduction. The degradation of amino acids involves deamination reactions that remove nitrogen-containing groups while channeling carbon skeletons into central metabolic pathways for energy production. Nucleotide metabolism supports the synthesis and maintenance of DNA and RNA, while providing energy currency and signaling molecules essential for cellular function. The synthesis of purine and pyrimidine nucleotides involves complex pathways that require substantial energy investment and careful regulation to maintain appropriate pools of these essential molecules. The degradation of nucleotides must handle the nitrogen-containing bases while recycling phosphate groups and ribose sugars for reuse in other metabolic processes. Metabolic regulation ensures that cellular metabolism responds appropriately to changing conditions while maintaining overall metabolic balance and avoiding futile cycles that would waste energy and resources. This regulation involves multiple mechanisms including allosteric enzyme regulation, covalent modification of enzymes, and transcriptional control of enzyme synthesis that operate on different timescales to provide both rapid responses and long-term adaptation. The integration of these regulatory mechanisms enables cells to coordinate metabolism with growth, division, and environmental responses. Enzyme kinetics and regulation represent crucial aspects of metabolic control that determine the rates of individual reactions and their responses to changing substrate concentrations and regulatory signals. Many metabolic enzymes exhibit complex kinetic behavior that includes allosteric regulation, where binding of regulatory molecules at sites distinct from the active site can dramatically alter enzyme activity. This regulatory mechanism enables metabolic pathways to respond sensitively to cellular conditions while maintaining stability and avoiding excessive fluctuations. Compartmentalization of metabolism within different cellular organelles enables eukaryotic cells to optimize different metabolic processes while maintaining appropriate concentrations of enzymes and substrates for efficient pathway function. The separation of fatty acid synthesis in the cytoplasm from fatty acid oxidation in mitochondria prevents futile cycling, while enabling independent regulation of these opposing pathways. Similarly, the compartmentalization of different aspects of nucleotide metabolism prevents interference among competing pathways while enabling specialized regulation. Metabolic flexibility enables cells to adapt to changing nutrient availability and energy demands by switching among different metabolic pathways and fuel sources. This flexibility involves both rapid regulatory responses that alter existing enzyme activities and longer-term responses that adjust enzyme levels through changes in gene expression. The ability to switch between glucose and fatty acid metabolism, for example, enables cells to maintain energy production during periods of nutrient limitation while optimizing metabolic efficiency. Alternative metabolic pathways provide backup systems and specialized functions that enable cells to respond to unusual conditions or perform specialized functions. The pentose phosphate pathway provides an alternative route for glucose metabolism that generates reducing power for biosynthesis while producing ribose sugars for nucleotide synthesis. Fermentation pathways enable energy production in the absence of oxygen, allowing cells to survive in anaerobic environments or during periods of oxygen limitation. Metabolic disorders demonstrate the critical importance of proper metabolic function and the consequences of metabolic disruption. Inherited defects in individual enzymes can cause severe metabolic diseases that highlight the essential roles of specific metabolic pathways in human health. Understanding these disorders has provided important insights into normal metabolic function, while driving the development of therapeutic approaches for metabolic diseases. The evolution of metabolism reveals how cellular energy systems have been refined and optimized over billions of years of evolutionary history. The conservation of core metabolic pathways across all domains of life suggests that these systems evolved early in the history of life and have been maintained due to their fundamental importance for cellular function. At the same time, the diversity of metabolic adaptations observed in different organisms demonstrates the flexibility and evolvability of metabolic systems. Chapter 4: Cell Division and Reproduction. Cell division represents one of the most fundamental and remarkable processes in biology, enabling the reproduction of cellular life while ensuring the accurate transmission of genetic information to daughter cells. This process must accomplish the extraordinary feat of duplicating all essential cellular components, including the complete genetic material, organelles, and molecular machinery, while distributing these components appropriately between two new cells that must each be capable of independent survival and function. The mechanisms that control cell division have evolved sophisticated regulatory systems that coordinate DNA replication, chromosome segregation, and cell separation to ensure the fidelity of cellular reproduction, while enabling the growth and development of multicellular organisms. The cell cycle represents the ordered sequence of events that leads from the formation of a cell through its growth and division to produce two daughter cells. This cycle is traditionally divided into distinct phases that reflect different cellular activities and molecular events. The G1 phase involves cellular growth and the accumulation of materials and energy necessary for DNA replication, while the S phase encompasses the complete duplication of the cellular genetic material. The G2 phase involves further growth and preparation for cell division, while the M phase includes the actual processes of chromosome segregation and cell division. DNA replication during S phase represents one of the most critical aspects of cell division, requiring the complete and accurate duplication of all genetic information contained within the cell. This process involves the unwinding of the double helix structure of DNA and the synthesis of new complementary strands using existing strands as templates. The molecular machinery responsible for DNA replication includes numerous enzymes and proteins that ensure high-fidelity copying while maintaining remarkable speed of synthesis. Multiple origins of replication enable the simultaneous initiation of DNA synthesis at many sites along chromosomes, allowing completion of replication within the limited time available during S phase. The accuracy of DNA replication is ensured by multiple proofreading and repair mechanisms that detect and correct errors that might arise during the synthesis process. DNA polymerases possess intrinsic proofreading activity that can detect and remove incorrectly incorporated nucleotides, while additional repair systems scan newly synthesized DNA for remaining errors. The overall error rate of DNA replication is remarkably low, typically less than 1 error per billion nucleotides incorporated, demonstrating the effectiveness of these quality control mechanisms. Chromosome condensation and organization during cell division involve the compaction of long DNA molecules into discrete structures that can be accurately segregated between daughter cells. This process requires the coordinated action of multiple proteins, including condensins that help organize and compact chromosomes, while ensuring that sister chromatids remain attached at centromere regions until the appropriate time for separation. The level of chromosome condensation increases dramatically during cell division, transforming the extended chromatin structure present during interphase into the highly compact chromosomes visible during mitosis. The mitotic spindle represents a remarkable molecular machine that accomplishes the precise segregation of chromosomes during cell division. This bipolar structure consists of microtubules emanating from centrosomes at opposite poles of the dividing cell, with some microtubules attaching to chromosomes at specialized structures called kinetochores, while others interact with microtubules from the opposite pole to maintain spindle integrity. The dynamic behavior of microtubules, involving continuous assembly and disassembly, enables both the capture of chromosomes and the generation of forces necessary for chromosome movement. Chromosome attachment and segregation involves sophisticated molecular mechanisms that ensure each daughter cell receives exactly one copy of every chromosome. Kinetochores represent complex protein structures that assemble at centromeres and provide attachment sites for spindle microtubules, while also serving as signaling platforms that monitor attachment status. The spindle checkpoint mechanism prevents cell division from proceeding until all chromosomes are properly attached to microtubules from both spindle poles, ensuring accurate chromosome segregation. Cytokinesis represents the physical process that divides the cellular cytoplasm and organelles between daughter cells while maintaining the integrity of cellular membranes and ensuring appropriate distribution of cellular components. In animal cells, this process involves the formation of a contractile ring composed of actin and myosin filaments that gradually constricts to pinch the cell into two separate entities. Plant cells face additional challenges due to their rigid cell walls, and accomplish cytokinesis through the formation of new cell wall material that gradually separates the daughter cells. Cell cycle checkpoints represent sophisticated regulatory mechanisms that monitor cellular conditions and ensure that cell division proceeds only when appropriate conditions are met. The G1S checkpoint evaluates whether cells are adequately prepared for DNA replication and have sufficient nutrients and energy for the process. The intra-S checkpoint monitors DNA replication progress and responds to replication problems by slowing or stopping the process until problems are resolved. The G2M checkpoint ensures that DNA replication is complete and that cells are ready for chromosome segregation. The molecular machinery controlling cell cycle progression involves cyclins and cyclin-dependent kinases that form regulatory complexes responsible for driving cells through different phases of the cycle. Cyclins are regulatory proteins whose levels fluctuate throughout the cell cycle, while cyclin-dependent kinases are enzymes that phosphorylate target proteins to promote cell cycle progression when activated by appropriate cyclins. The activity of these complexes is further regulated by inhibitory proteins and phosphatases that can prevent cell cycle progression under inappropriate conditions. P53 represents one of the most important tumor suppressor proteins that monitors cellular stress and can halt cell cycle progression or trigger cell death when problems are detected. This protein responds to various stress signals including DNA damage, hypoxia, and oncogene activation by activating transcriptional programs that can lead to cell cycle arrest, DNA repair, or apoptosis. The critical importance of P53 in preventing cancer development is demonstrated by the fact that this gene is mutated in approximately half of all human cancers. Apoptosis represents a form of programmed cell death that enables the elimination of damaged or unnecessary cells while maintaining tissue homeostasis. This process involves a coordinated series of cellular changes, including chromosome condensation, membrane blebbing, and cellular fragmentation that facilitate the removal of dying cells without triggering inflammatory responses. The decision to undergo apoptosis is controlled by complex signaling pathways that evaluate cellular stress levels and environmental conditions. Stem cells represent specialized cell populations that maintain the ability to undergo unlimited cell division, while retaining the capacity to differentiate into various specialized cell types. These cells play crucial roles in development, tissue maintenance and repair throughout the life of multicellular organisms. The regulation of stem cell division involves unique mechanisms that balance self-renewal with differentiation to maintain appropriate stem cell populations while providing specialized cells as needed. Cell division in prokaryotic organisms involves simpler mechanisms than those found in eukaryotic cells, but still requires sophisticated coordination to ensure accurate DNA replication and chromosome segregation. Binary fission represents the primary mode of prokaryotic reproduction, involving DNA replication, followed by cell elongation and division that produces two identical daughter cells. Despite the apparent simplicity of this process, it requires precise coordination of DNA replication, chromosome segregation and cell wall synthesis. Asymmetric cell division represents a specialized form of cell division, where daughter cells inherit different cellular components or developmental fates, enabling the generation of cellular diversity within developing organisms. This process involves the asymmetric distribution of regulatory proteins, organelles, or other cellular components that influence the subsequent development of daughter cells. Asymmetric division plays crucial roles in development and stem cell biology by generating cellular diversity while maintaining stem cell populations. Meiosis represents a specialized form of cell division that produces gametes for sexual reproduction while reducing chromosome number from diploid to haploid. This process involves two successive divisions with unique features including chromosome pairing, genetic recombination and reduction division that create genetically diverse gametes essential for sexual reproduction. The complexity of meiosis reflects the additional challenges of managing paired chromosomes while ensuring accurate segregation and genetic diversity. The evolution of cell division mechanisms reveals how these fundamental processes have been refined and optimized throughout the history of life. The conservation of core cell division machinery across diverse organisms suggests that these mechanisms evolved early and have been maintained due to their essential roles in cellular reproduction. At the same time, variations in cell division mechanisms among different organisms demonstrate the adaptability of these systems to different cellular and organismal requirements. Chapter 5: Cell Communication and Response Systems. Cell communication represents one of the most sophisticated and essential aspects of cellular biology, enabling individual cells to coordinate their activities with neighboring cells and respond appropriately to environmental changes and internal conditions. This complex process involves the detection of chemical, physical, and electrical signals through specialized receptor proteins, the transmission of these signals through intracellular signaling pathways, and the generation of appropriate cellular responses that can range from changes in gene expression to alterations in cellular behavior and metabolism. Understanding cell communication requires examining both the molecular mechanisms that enable signal detection and transmission, and the integrated systems that allow cells to process multiple simultaneous signals while generating coordinated responses. Signal transduction represents the fundamental process by which cells detect, process, and respond to information from their environment and from other cells. This process typically begins with the binding of signaling molecules to specific receptor proteins that undergo conformational changes upon ligand binding, triggering intracellular signaling cascades that amplify and transmit the signal throughout the cell. The specificity of signal transduction depends on the precise molecular recognition between signaling molecules and their receptors, while the sensitivity of these systems enables cells to respond to extremely low concentrations of signaling molecules. Receptor proteins represent the molecular gatekeepers that determine which signals cells can detect and how they respond to these signals. These proteins exhibit remarkable diversity in structure and function, with different receptor types specialized for detecting specific classes of signaling molecules including hormones, neurotransmitters, growth factors, and environmental stimuli. Cell surface receptors detect signals that cannot cross cellular membranes, while intracellular receptors bind to signaling molecules that can diffuse through cellular membranes to reach their targets within cells. G-protein coupled receptors represent one of the largest and most important families of cell surface receptors, detecting an enormous variety of signaling molecules including light, odors, tastes, hormones, and neurotransmitters. These receptors share a characteristic 7-transmembrane domain structure that undergoes conformational changes upon ligand binding, leading to the activation of associated G proteins that initiate intracellular signaling cascades. The diversity of G-protein coupled receptors and their associated signaling pathways enables cells to respond to virtually any conceivable external signal. Enzyme-linked receptors represent another major class of cell surface receptors that possess intrinsic enzymatic activity or are associated with enzymes that become activated upon receptor binding. Receptor tyrosine kinases exemplify this class of receptors, undergoing dimerization and autophosphorylation upon growth factor binding, creating docking sites for intracellular signaling proteins that initiate complex signaling networks. These receptors play crucial roles in controlling cellular growth, differentiation, and survival in response to extracellular signals. Ion channel receptors enable rapid cellular responses to extracellular signals through the direct control of ion flow across cellular membranes. These receptors undergo conformational changes upon ligand binding that open or close ion channels, leading to immediate changes in membrane potential and cellular excitability. The speed of ion channel responses makes them particularly important for rapid communication in the nervous system, where rapid changes in membrane potential enable the transmission of electrical signals over long distances. Intracellular signaling pathways represent complex networks of molecular interactions that transmit and amplify signals from receptors to their ultimate cellular targets. These pathways often involve protein kinase cascades, where sequential phosphorylation events amplify signals, while providing multiple points for regulation and integration with other signaling pathways. The modular organization of signaling pathways enables cells to mix and match different signaling components to create diverse responses to different stimuli. Second messenger systems provide mechanisms for signal amplification and distribution throughout cells, enabling small numbers of activated receptors to generate large cellular responses. Cyclic adenosine monophosphate represents one of the most important second messengers, generated by adenylyl cyclase in response to G-protein activation, and capable of activating protein kinase A to phosphorylate numerous target proteins. Calcium ions represent another crucial second messenger system, with carefully regulated calcium concentrations enabling precise control of numerous cellular processes. Protein kinases and phosphatases represent key regulatory enzymes that control cellular responses through the phosphorylation and dephosphorylation of target proteins. These enzymes can rapidly and reversibly modify protein function, enabling dynamic regulation of cellular processes in response to signaling pathways. The human genome encodes hundreds of different protein kinases and phosphatases, reflecting the central importance of phosphorylation-based regulation in cellular signaling. Transcriptional regulation represents a major mechanism through which signaling pathways can produce long-lasting changes in cellular behavior by altering gene expression patterns. Many signaling pathways ultimately lead to the activation or inhibition of transcription factors that control the expression of specific sets of genes. These transcriptional responses enable cells to adapt to new conditions by producing new proteins or altering the levels of existing proteins to meet changing cellular requirements. Cell-cell communication involves direct physical contacts between adjacent cells through specialized intercellular junctions that enable the transfer of small molecules and electrical signals. Gap junctions represent channels formed by connexin proteins that directly connect the cytoplasms of adjacent cells, enabling the rapid equilibration of ions, metabolites, and signaling molecules. This direct communication enables groups of cells to coordinate their activities and respond collectively to stimuli. Paracrine signaling involves the release of signaling molecules that act on nearby cells within local tissue environments. This type of communication enables groups of cells to coordinate their activities while maintaining relatively local effects that do not influence distant tissues. Growth factors, cytokines, and many other signaling molecules function through paracrine mechanisms to control tissue development, immune responses, and tissue repair processes. Endocrine signaling involves the release of hormones into the circulatory system, where they can travel throughout the body to influence distant target tissues. This type of communication enables the coordination of physiological processes across different organ systems and provides mechanisms for systemic responses to environmental changes. The specificity of endocrine signaling depends on the expression of appropriate hormone receptors in target tissues. Autocrine signaling involves cells producing signaling molecules that act on the same cells that produce them, creating feedback loops that can amplify or modulate cellular responses. This type of signaling plays important roles in development, immune responses, and cancer, where cells can stimulate their own growth and survival through autocrine loops. The regulation of autocrine signaling requires sophisticated mechanisms to prevent excessive self-stimulation. Signal integration represents the remarkable ability of cells to process multiple simultaneous signals and generate appropriate responses that reflect the combined input from different signaling pathways. This integration occurs through various mechanisms including shared signaling components, cross-regulation among pathways, and combinatorial transcriptional control that enables cells to generate diverse responses to different combinations of signals. The ability to integrate multiple signals enables cells to make complex decisions about their behavior based on comprehensive information about their environment. Adaptation and desensitization mechanisms enable cells to adjust their sensitivity to signaling molecules over time, preventing excessive responses to persistent stimuli while maintaining responsiveness to changes in signal levels. These mechanisms involve feedback regulation of receptor expression, receptor modification, and downstream signaling components that reduce cellular responses during prolonged stimulation. Adaptation enables cells to respond to changes in their environment rather than absolute signal levels. Signaling in development involves the use of cell communication systems to coordinate the complex processes of embryonic development, where cells must differentiate into specialized types while organizing into complex tissue and organ structures. Morphogen gradients provide positional information that enables cells to determine their location within developing embryos and adopt appropriate developmental fates. The precision of developmental signaling requires sophisticated mechanisms for generating and interpreting concentration gradients of signaling molecules. Pathological signaling demonstrates the critical importance of proper cell communication for health and the consequences of signaling dysfunction. Cancer often involves disruption of normal growth control signaling, while autoimmune diseases involve inappropriate immune signaling responses. Understanding pathological signaling has provided important insights into normal signaling mechanisms, while driving the development of therapeutic approaches that target specific signaling pathways.